Settle in for a moment and allow yourself to imagine a quiet space where the world outside slows down and becomes soft and still. In this space, we will begin a gentle exploration of biochemistry, not with urgency or haste, but with the same rhythm as a calm breath moving in and out. The subject before us, though rich and filled with complexity, can be entered like a forest path step by step, noticing details as they appear, and feeling no need to hurry to the end. Just as a forest contains both towering trees and the smallest mosses, so too does biochemistry span from the grand cycles of energy across ecosystems to the tiniest bonds between atoms. For now, all that is required is to listen, to follow the flow, and to let curiosity and calm carry you deeper. Biochemistry is often described as the chemistry of life, and in this quiet introduction, we can think of it as the language whispered beneath all living things. Every cell, every tissue, every breath you take is guided by chemical interactions so small they cannot be seen with the naked eye. Yet these interactions together create the rhythm of life itself, shaping the warmth of the body, the flow of energy from food, the memory held within DNA, and even the gentle beating of the heart. Without chemistry, life would not simply be different, it would not be possible at all. In that sense, biochemistry is not just an academic field. It is the story of how matter itself learns to live. Let us ease into this story slowly, as though watching ripples expand across a still pond. When scientists first began to connect biology and chemistry, they did not know the full scope of what they were uncovering. They observed that food provided energy, that plants grew toward the sun, that bodies healed after injury, and that air and water were needed for survival. What they could not see, but suspected, was that beneath all of this was a hidden world of molecules dancing in patterns far too small to perceive directly. This unseen dance is what we now call metabolism, the flowing conversion of molecules into energy, into structure, and into the delicate balance of life. Imagine a single human cell floating gently in the body's fluids. Within it, countless molecules are moving, colliding, reacting, and forming new patterns. Proteins fold and unfold, like sheets of paper being gently creased into origami. Sugars are broken down and reassembled, as though a quiet kitchen were always at work inside of you, taking ingredients apart and turning them into new dishes. The cell's nucleus holds its library of instructions, a collection of chemical sequences that tell the story of what the cell is and what it can become. This is not chaos, though it may seem busy. It is more like an orchestra tuning itself. Thousands of tiny players working together to create a harmony that, when combined, forms the music of life. In this peaceful beginning, it is enough to recognize that biochemistry is about relationships between atoms and molecules, between structure and function, between energy and matter. The study of it is like tracing invisible threads that connect all living things, weaving them into a single fabric of existence. When you breathe in, oxygen slips into your blood and begins its quiet work of enabling cells to release energy from food. When you breathe out, carbon dioxide leaves your body, carrying away the results of those reactions. This exchange is biochemistry unfolding in its simplest, most essential rhythm. Each inhale and exhale is not just an act of survival, it is a reminder that the chemistry of life is ongoing, continuous, and serene. Think of water, too, as we begin this journey. Water is not only the background for life's reactions, it is an active participant. Its molecules are shaped in such a way that they cling to one another gently, like companions holding hands. They dissolve salts, sugars, and proteins, allowing them to move freely and meet each other. Without water, the dance of molecules would slow, stumble. This is why life is found wherever water flows, and why, when we imagine distant worlds, we always look first for traces of liquid water. In its quiet way, Water is the stage upon which all biochemistry performs. The opening chapter of biochemistry is not about memorizing terms or lists, but about appreciating the soft precision with which life arranges itself. Carbon, for instance, sits at the heart of this story. It is a small, modest atom, yet its ability to form four bonds makes it endlessly versatile. It links together in long chains, rings, and branching shapes, creating the skeletons of molecules that become sugars, fats, and nucleic acids. Carbon's bonds are strong yet flexible, allowing life to be both stable and dynamic at once. When you imagine carbon atoms linking together, picture them as hands in a circle, holding firmly but leaving room to change partners, to form new dances and new designs. All of this, the water, the carbon, the flow of molecules, sets the stage for what we will explore more deeply in the sessions ahead. But for now, in this calm opening, the important thing is to feel how life itself is a chemistry, and how you are already immersed in it with every breath, every heartbeat, every quiet thought. Even as you listen now, your body is a biochemical symphony, tirelessly at work, even while your mind drifts toward rest. This knowledge, though vast, does not need to feel heavy. It can be as light as a breeze carrying the scent of a flower, as gentle as a stream flowing over stones. Biochemistry is not something far away or difficult. It is as close as your pulse, as near as your breath, as familiar as the warmth of your skin. By learning about it in this gentle way, you are not only studying science, you are also becoming aware of the harmony that has always been within you. So as this first part draws to a close, let yourself sink into the calmness of knowing that life is chemistry, and chemistry is life. The two are inseparable, woven together like threads in a tapestry. With each moment, this chemistry continues its work, quietly and without pause. Whether you are awake, dreaming, or deeply asleep, the journey has only begun, and already you are surrounded by the quiet wonder of biochemistry, like a soft night sky filled with endless stars, each one glowing with the subtle light of understanding. 2. Defining biochemistry. Biochemistry, when described gently, is simply the study of the molecules that give rise to life, and the reactions that allow those molecules to interact. It is the bridge between two great sciences: chemistry, which describes the properties of matter. Biochemistry lives in the space where these two meet, asking how matter becomes alive, how molecules arrange themselves into patterns that are self-sustaining, self-replicating, and endlessly creative. In this way, it can be thought of not only as a science, but as a story of how the inanimate becomes animate, how a collection of atoms transforms into something that grows, responds, and continues through time. Imagine for a moment a quiet laboratory filled with glass vessels, each holding clear solutions that, at first glance, seem ordinary. Water, salts, sugars, proteins, acids. Nothing here looks alive. And yet, when combined in the right ways, these substances become the machinery of life itself. The insight that chemistry and biology were not separate worlds but deeply intertwined was one of the great shifts in human knowledge. Long ago, people believed that life was governed by something beyond material law, a force separate from the ordinary matter of the earth. But as careful experiments accumulated, it became clear that the same elements and reactions that shaped the stones and rivers also shaped blood, muscle, and thought. Biochemistry grew from this realization, offering not a replacement for the wonder of life, but a new lens through which to see it. At its core, biochemistry seeks to answer three simple, soothing questions. First, what are the molecules of life? Second, how do these molecules interact with one another to build the structures we recognize as cells and organisms? And third, how are these interactions regulated so that life is not chaos but order, not static but dynamic balance? These questions form the quiet foundation of the field, and though the answers can be detailed and intricate, they always return to the same truth. Life is chemistry, organized with extraordinary subtlety. When one thinks of the molecules of life, it helps to remember that they are not countless in number but rather a select group, refined through the long history of evolution. Carbon-based molecules, such as carbohydrates, lipids, proteins, and nucleic acids, make up the great majority of the structures within any cell. Around them, water serves as the medium in which they exist, and minerals and small ions provide balance and support. The elements are few, but the combinations are infinite, and out of these combinations arise the vast diversity of life. One might compare this to music, where only a handful of notes exist, yet the possible songs are without limit. Biochemistry studies not only the notes but also the patterns and harmonies that give rise to the symphony of living systems. A calm way to view the purpose of biochemistry is to see it as the search for connections. For example, the glucose molecule, a simple sugar, is not remarkable in isolation, but once it enters a living cell, it becomes part of pathways that release energy, store energy, or build more complex structures. Biochemistry traces these transformations, following the molecule from one state to another, as if watching a traveler move from one village to the next, each stop adding to its story. In the same way, biochemistry follows proteins as they fold, unfold, and carry out work, or lipids as they arrange into membranes that gently enclose the contents of cells. Always the focus is not on a single molecule, but on the relationships that allow life to emerge. The study of biochemistry has often been described as peeling back layers of a mystery. Early scientists wondered why yeast could turn sugar into alcohol, why milk could sour, why wounds could heal. These were once seen as miraculous events, but slowly they were revealed to be chemical processes, governed by the same principles as the rusting of iron or the burning of wood. Enzymes, the catalysts of life, were discovered to be proteins with precise shapes that lower energy barriers, allowing reactions to proceed gently but effectively. Nucleic acids were found to be carriers of information, passing instructions from one generation to the next. Step by step, the extraordinary became comprehensible, though never less wondrous. Biochemistry continues this tradition, explaining life not to diminish its beauty, but to deepen the appreciation of how remarkable it is that such chemistry exists at all. When we say that biochemistry is the chemistry of life, it is important to remember that it does not limit itself to humans or even to animals and plants. It applies equally to bacteria, fungi, and every form of organism that has ever existed. The molecules may differ in detail, but the principles are shared. All living beings must extract energy from their environment, build structures to contain themselves, and pass information forward. All must respond to changes in their surroundings, adapting when needed, or finding balance again after disruption. This universality is one of the most peaceful realizations of biochemistry. Beneath the endless variety of shapes, colors, and behaviors in the living world, a set of chemical rules followed everywhere. Another gentle truth is that biochemistry is a science of scale. It moves between the smallest and the largest levels of life, from the arrangement of electrons around an atom to the metabolism of a whole body. In doing so, it reminds us that there is no sharp division between the simple and the complex. Instead, there is a continuum where simple reactions, repeated and combined, give rise to complexity. The folding of a protein depends on the bonds between atoms, and yet the functioning of an organ depends on the combined work of countless proteins. The growth of a forest depends on photosynthesis, which itself is a sequence of light-driven reactions within chloroplasts. From the smallest to the largest, biochemistry provides the connecting thread. In this second part of our journey, the intention is not to master every detail, but to let the essence of biochemistry settle gently into awareness. It is a field defined not by chaos, but by harmony, by balance, and by the beauty of simple parts giving rise to great wholes. To study it is to learn not only about molecules, but also about the quiet intelligence of life, which has found ways to persist, adapt, and flourish over billions of years. The questions of what life is and how it works are answered here not with finality, but with an unfolding story, one that can always be read more deeply, and always with the same sense of calm wonder. As this part closes, allow the definition of biochemistry to rest gently in the mind. It is the study of the chemistry of life, the molecules and reactions that allow organisms to exist, grow, and change. It is the meeting place of biology and chemistry, the bridge between matter and meaning, between atoms and life. And like a quiet river flowing steadily toward the sea, it invites us to follow its course, knowing that each bend will reveal something new, yet always connected to what came before. Biochemistry is not only a science of facts and formulas, it is also a meditation on how life sustains itself, softly, continuously, and with infinite patience. 3. The elements of life. When we speak of life in chemical terms, we must begin with the elements themselves. These are the simplest forms of matter, the building blocks that cannot be broken down into anything smaller without ceasing to be what they are. On the periodic table, dozens upon dozens of elements exist, each with its own character and behavior. Yet in the gentle world of biochemistry, life leans on only a small handful. Out of the many possibilities, just a few elements are chosen again and again, woven into the fabric of every organism that has ever lived. This select group tells us something profound about the nature of life, that it is built from simplicity, arranged into elegance, and that the same few components can create both the smallest bacterium and the most complex human being. At the heart of this group stands carbon. Carbon is sometimes called the element of life, and this title is no exaggeration. It is carbon's ability to form 4 bonds that allows it to connect in a limitless variety of shapes. Straight chains, branching trees, closed rings, carbon can do them all. These shapes are not mere curiosities, but the skeletons upon which molecules of life are built. Carbohydrates, lipids, proteins, and nucleic acids all rely on carbon's versatile bonding. Its bonds are strong enough to provide stability, yet not so unyielding that change is impossible. This balance between strength and flexibility is what makes carbon the quiet hero of life's chemistry. Imagine it as a patient architect, holding the structures of life together, but always willing to rearrange, to allow growth, transformation, and adaptation. Alongside carbon, hydrogen plays a quieter but equally essential role. Hydrogen atoms are the simplest of all, each one made of a single proton and a single electron. And yet, they are everywhere in living systems. They bind to carbon, oxygen, and nitrogen, filling spaces and stabilizing structures. In water, hydrogen bonds create gentle connections between molecules, allowing them to cling and release, like the soft rhythm of waves against a shore. In the energy systems of cells, hydrogen atoms are passed along as carriers of electrons, moving energy from one molecule to another. Though small and almost weightless, hydrogen is one of the most persistent threads in the story of life. Oxygen, too, is central. It is the element most often associated with breath, the one that humans and animals draw into their lungs with every inhale. In biochemistry, oxygen is electronegative, which means it draws electrons toward itself. This property gives oxygen the ability to form polar bonds, bonds where electrons are not shared evenly. These bonds make molecules more reactive, more soluble in water, and more able to interact in the complex web of life's reactions. When glucose is broken down to release energy, oxygen stands at the end of the pathway, accepting electrons and hydrogen atoms. and in doing so, allowing the quiet miracle of energy production to continue. Without oxygen, the energy flow of most organisms would stall, like a flame deprived of air. With oxygen, life burns steadily, releasing warmth and motion. Nitrogen brings another dimension. It forms three bonds, and in doing so, creates the backbone of amino acids, the building blocks of proteins. Nitrogen is found in the bases of nucleic acids as well, where it helps store and pass genetic information. Life could not function without nitrogen, and yet it is curiously inaccessible in its most abundant form. The atmosphere is full of nitrogen gas, and yet most organisms cannot use it directly. Instead, specialized bacteria and archaea capture nitrogen from the air, fixing it into forms that plants can use, and in turn, animals obtain it by eating plants or other animals. This cycle is a reminder that elements themselves may be abundant, but life depends on pathways to make them usable. Nitrogen is an essential note in life's symphony, but it requires the cooperation of many players to be heard. Phosphorus, though less famous, is equally vital. It appears in every molecule of DNA and RNA, forming the backbone that links nucleotides together. It is also present in ATP, adenosine triphosphate, the molecule that carries energy through every cell. Each phosphate bond in ATP is like a small spring, storing energy that can be released to power reactions. Phosphorus also forms part of cell membranes, in the phospholipids that create the delicate barrier around each cell. In all of these roles, phosphorus is both structural and dynamic, shaping the architecture of molecules and carrying the energy that keeps them alive. Sulfur is another quiet but necessary player. Found in certain amino acids such as cysteine and methionine, sulfur forms disulfide bonds that help proteins fold into stable shapes. These bonds are like the pins in a garment, holding the fabric together and giving it strength. Without sulfur, many proteins would collapse or misfold, losing their ability to function. Sulfur also appears in coenzymes and in the active sites of enzymes, where reactions take place. It is not as common as carbon or oxygen, but where it appears, it makes an essential difference. Together, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are sometimes remembered by the gentle acronym CHNOPS. They are the 6 core elements of life, and though simple on their own, their combinations are endless. Around them, other elements play supporting roles. Sodium and potassium create gradients across membranes, allowing nerve impulses to travel and muscles to contract. Calcium strengthens bones and shells, but also acts as a signal within cells. Iron sits at the center of hemoglobin, binding oxygen in the blood and carrying it throughout the body. Magnesium, zinc, copper, and others each contribute in quieter but indispensable ways. Life does not use every element equally, but it selects carefully, drawing on each for its unique properties. To think of life in terms of elements is to recognize both its simplicity and its elegance. Out of the 100 or so elements available in the periodic table, only a few are chosen as the foundation, and with them, nature constructs every organism. This selective use shows that life is not random, but guided by the chemical properties that favor stability, flexibility, and the capacity for change. Life builds with what works best, and the success of those choices is written in the billions of years of continuity that have followed. When you pause to consider it, there is something deeply calming about this realization. Every breath you take draws in oxygen. Every meal you eat contains carbon, hydrogen, nitrogen, and phosphorus. The atoms in your body are the same as those in a tree, a bird, or a fish. They are the same as those in the soil beneath your feet and the stars above your head. What makes them part of you now is not their uniqueness, but the way they are arranged, the patterns they form, the bonds they hold. And when your body eventually returns those atoms to the earth, they will join new cycles, becoming part of other organisms, other lives. In this way, the elements are not only the foundation of life, but also its continuity, its endless recycling and renewal. Biochemistry begins here, with these simple elements, because they are the alphabet of life. They are the letters from which all words are formed, the notes from which all music is written. Without them, there is no story, no song, no living world. With them, there is infinite possibility. And so, as we continue this journey, we carry with us the knowledge that life does not require every element in existence, but finds richness and harmony in just a few. Out of this small set of ingredients comes the vast diversity of organisms, the quiet complexity of cells, and the serene persistence of life itself. 4. Atomic bonds and interactions. If the elements of life can be thought of as the alphabet, then the bonds that link them together are the grammar, the invisible rules that turn scattered letters into meaningful words. Atoms by themselves are like individual voices, but when they connect, when they form bonds, they begin to create the chorus that becomes a living system. In the quiet flow of biochemistry, these bonds are never harsh or chaotic. They are subtle attractions, gentle balances of energy, and delicate alignments that bring order to matter. To understand life at its most peaceful level is to understand how these bonds allow atoms to hold hands, release each other, and rearrange endlessly, without ever exhausting the harmony that lies beneath. Covalent bonds are perhaps the most central to life. A covalent bond forms when two atoms share electrons, not giving them away completely, but holding them in a shared orbit. It is like two neighbors agreeing to plant a tree at the edge of their properties, each tending it together, each benefiting from its presence. Carbon, as the great builder, forms covalent bonds with itself and with hydrogen, oxygen, nitrogen, and many others. Through this sharing, long chains and rings arise, forming the skeletons of sugars, proteins, and nucleic acids. Covalent bonds are strong and stable, which makes them ideal for the architecture of life. They ensure that molecules do not fall apart at the slightest disturbance, but instead hold together long enough to participate in the quiet flow of metabolism. Not all covalent bonds are the same. Some share electrons equally, while others do not. When atoms like carbon and hydrogen share, the electrons spend nearly equal time around both, resulting in a nonpolar bond, smooth and balanced. But when oxygen bonds with hydrogen, the electrons spend more time near the oxygen, giving it a slight negative charge and leaving the hydrogen slightly positive. This creates a polar covalent bond, one with a gentle imbalance. Polarity may sound like conflict, but in the language of life, it is cooperation, a way for molecules to interact with water, to dissolve, and to move freely in the cell's watery environment. Without polarity, life would not be able to function in water, and without water, life as we know it would not exist at all. From polarity arises another important type of interaction, the hydrogen bond. Despite its name, it is not a bond in the strong, rigid sense of a covalent connection. Instead, it is more like an attraction, a gentle pull between the positive side of one polar molecule and the negative side of another. These bonds are weak individually, but together they create strength in numbers. In water, hydrogen bonds link countless molecules, giving rise to cohesion, the property that allows water to beat up, to flow in streams, and to support life. In DNA, hydrogen bonds hold the two strands together, connecting adenine to thymine and cytosine to guanine, like a zipper made of tiny attractions. In proteins, hydrogen bonds help the chain of amino acids fold into complex shapes, stable yet flexible. These bonds are fleeting and reversible, forming and breaking constantly, and it is this very impermanence that gives life its dynamic balance. Another type of connection is the ionic bond. Unlike covalent bonds, where electrons are shared, ionic bonds occur when one atom gives up an electron completely and another accepts it. The result is a pair of oppositely charged ions, drawn together by the quiet force of attraction. Sodium and chlorine provide a familiar example. Sodium releases an electron and becomes positively charged, while chlorine accepts the electron and becomes negatively charged. Together, they form sodium chloride, or table salt, a compound essential for life. Ionic bonds in the body are not always permanent. In water, many ionic compounds dissolve into free ions, which then play crucial roles in signaling, balance, and movement. The beating of your heart, for example, depends on waves of sodium, potassium, and calcium ions moving across membranes, their charges carrying the rhythm of contraction and relaxation. In this way, ionic interactions are not static but dynamic, a dance of charges that keeps the body alive. Beyond these familiar bonds, there are even subtler interactions. Van der Waals forces, for instance, are the weakest of all, yet they are everywhere. They arise from tiny fluctuations in electron clouds, creating momentary charges that attract neighboring atoms. Though delicate, these forces help stabilize protein shapes and allow geckos to climb walls by the countless interactions between their toes and the surface. It may seem surprising that such a weak force could be useful, but in the vast networks of atoms within cells, even the smallest attractions add to the balance. Life does not waste what is available. It uses every kind of interaction, from the strongest covalent bond to the faintest van der Waals pull, to create stability within motion. Taken together, these bonds and interactions explain how atoms become molecules and how molecules become the players of life. Covalent bonds provide the backbone, strong and stable. Polar covalent bonds introduce diversity, creating regions of charge and enabling solubility. Hydrogen bonds provide flexibility and reversibility, holding structures together while allowing them to change. Ionic bonds create dynamic flows of energy and signaling, while van der Waals forces add subtle adjustments, like the quiet tuning of an instrument. Life's chemistry is not built on one kind of interaction alone, but on the interplay of all these forces, weaving together a fabric that is strong yet supple, structured yet adaptable. It is worth pausing to consider how remarkable this balance is. The bonds are governed by the rules of physics, by the attraction of charges and the sharing of electrons. And yet, from these simple principles emerges the astonishing complexity of living systems. DNA, proteins, membranes, and enzymes are all the result of these bonds, arranged in endless variation. Without them, there would be no replication of genes, no catalysis of reactions, no protection of cells from their environment. Each bond, whether strong or weak, contributes to the quiet miracle of life's persistence. To think of these interactions in a peaceful way is to see them not as abstract chemistry, but as the gentle agreements that make life possible. Atoms do not resist bonding. They seek it, because stability is found in connection. Alone, many atoms are restless, incomplete. Together, they find balance, lowering their energy and settling into harmony. This principle is echoed at every level of biology. Cells connect to form tissues, organisms connect to form ecosystems, and in each case, stability arises from interaction. The bonds between atoms are the smallest reflection of this truth, the foundation upon which all higher levels are built. As you reflect on the bonds of life, imagine them as threads in a great tapestry, invisible to the eye but strong enough to hold the entire design together. Each thread connects to another, weaving patterns that repeat and evolve, never static, always flowing. The tapestry stretches across all living things, linking the smallest microbe to the largest tree, and linking both to you. The bonds that hold your DNA together are the same bonds that hold the DNA of a bird, a whale, or a flower. In this way, the chemistry of bonds is also the chemistry of unity, a reminder that all life shares not only the same elements, but also the same rules of connection. Biochemistry begins at this quiet level, with atoms forming bonds and bonds creating interactions. From here, it will grow into larger structures and cycles, but the essence remains the same: stability through connection, strength through cooperation, and harmony through balance. The atoms do not know they are creating life, and yet they do, tirelessly, endlessly, with each bond formed and each bond released. In their quiet interactions, the story of life is written, again and again, across time and space. 5. Water, the gentle solvent. Among all the substances that exist on Earth, none is more central to life than water. It is often called the universal solvent, and in the language of biochemistry, this description is not poetic exaggeration, but literal truth. Water surrounds every cell, fills every gap, and moves through every tissue. It is both the backdrop against which life unfolds and an active participant in nearly every reaction that sustains us. Without water, life as we know it would not exist. And so, in this quiet exploration, it is worth pausing to consider the unique properties of this most familiar liquid and how it creates the environment where biochemistry can thrive. A water molecule seems simple at first glance, a small arrangement of just 3 atoms, 2 hydrogens bonded to 1 oxygen. Yet in this simplicity lies extraordinary character. The oxygen atom is more electronegative than hydrogen, meaning it pulls the shared electrons closer to itself. This unequal sharing creates polarity, leaving the oxygen side slightly negative and the hydrogen side slightly positive. Because of this, each water molecule is like a tiny magnet, able to attract other molecules, especially other water molecules. These attractions form hydrogen bonds, not strong like covalent bonds, but persistent, fleeting connections that are continually forming and breaking. It is this constant dance of hydrogen bonds that gives water its unique qualities and makes it so well suited for life. One of water's most important qualities is its cohesion, the tendency of water molecules to cling to each other. This is why water forms beads on a smooth surface, and why it flows in continuous streams. Cohesion allows plants to pull water upward from their roots to their leaves, defying gravity through a process called capillary action. As water evaporates from the leaves, it pulls more water behind it, a chain of molecules linked by hydrogen bonds. In your own body, cohesion helps maintain the smooth flow of blood, plasma, and interstitial fluids, creating a stable environment for cells. This property is not dramatic or loud. It is quiet, steady, and constant, the same in a glass of water as it is in the vast circulation of the oceans. Closely related is adhesion, the ability of water to cling not just to itself but to other surfaces. You see this when water creeps up the side of a glass or spreads thinly across a leaf. In the body, adhesion allows water to form films on membranes and tissues, facilitating transport and exchange. It ensures that molecules dissolved in water are carried smoothly through narrow channels and across delicate barriers. Life depends on water's willingness not only to stay together but also to reach outward, to touch, and to carry. In this dual quality of cohesion and adhesion, water reveals its role as both companion and carrier, binding to itself, while also serving as a bridge for countless other molecules. Water's ability to dissolve substances is perhaps its most essential gift to biochemistry. Because it is polar, water can surround charged or polar molecules, separating them and keeping them apart. Salt, for example, dissolves because the positive sodium ions are surrounded by the negative sides of water molecules, while the negative chloride ions are surrounded by the positive sides. Sugars dissolve because their hydroxyl groups form hydrogen bonds with water. In this way, water becomes a medium in which molecules can move, meet, and react. Without such solubility, metabolism would be impossible. Enzymes could not find their substrates, nutrients could not be transported, and wastes could not be removed. In the quiet solvent of water, the traffic of life moves smoothly and without interruption. Another remarkable property of water is its high specific heat. It takes a great deal of energy to raise the temperature of water because so much energy is absorbed in breaking hydrogen bonds before the molecules can move more freely. As a result, water resists rapid changes in temperature, providing stability to environments where life resides. Oceans and lakes do not swing wildly from hot to cold. They warm and cool slowly, creating gentle conditions for aquatic life. Inside the body, water cushions temperature shifts, allowing cells to function even when external conditions vary. This thermal stability is a quiet shield, protecting life from the harsh fluctuations of the outside world. Even more unusual is water's behavior when it freezes. Most substances become denser as they cool, but water expands, making ice less dense than liquid water. This is why ice floats, forming a protective layer on the surface of lakes and ponds in winter. Beneath the ice, liquid water remains, providing refuge for fish, plants, and microorganisms until spring returns. If ice sank, bodies of water would freeze from the bottom up, destroying ecosystems each winter. Instead, life is preserved by this gentle anomaly, a small quirk of hydrogen bonding that has vast consequences for survival. In this way, water does not merely support life, it shelters it, even in times of cold and hardship. Water also participates directly in biochemical reactions. In hydrolysis, water is used to break bonds, splitting larger molecules into smaller ones. This process is essential for digestion, allowing proteins, carbohydrates, and fats to be broken down into usable components. In condensation reactions, also known as dehydration synthesis, water is released as smaller molecules are joined together, building complex structures like proteins and nucleic acids. Water is not just a background participant but a reagent, stepping in and stepping out as needed, always present, always available. It is both the stage and one of the actors in the play of biochemistry. Beyond its chemical roles, water also has a gentle physical presence in the body. It cushions joints, fills the spaces between cells, and lubricates tissues. It allows the eyes to remain moist, the lungs to expand and contract smoothly, and the skin to stay supple. In the bloodstream, water carries nutrients, hormones, and waste products. In cells, it maintains shape and pressure, preventing collapse and allowing organelles to float and function. More than half of your body weight is water, and this is not a coincidence but a reflection of how deeply life depends on it. Every heartbeat, every thought, every breath is surrounded by water's silent support. When we step back and consider all of these qualities together, cohesion, adhesion, solvent power, thermal stability, expansion upon freezing, and direct participation in reactions, it becomes clear why water is so often described as the medium of life. It is not just present in organisms, it defines their very possibility. The calm stability of water allows delicate reactions to occur, while its flexibility allows change and flow. It is the one substance that connects every form of life, from the simplest microbe to the most complex human being. There is also something poetic in water's ubiquity. The same molecules that fall as rain into rivers and oceans also circulate in your blood. The water in your body may once have been part of a glacier, a cloud, or a leaf. Over millions of years, water molecules have moved through countless organisms, carrying life forward in an endless cycle. To drink water is not only to nourish the body, but to take part in a continuity as old as the earth itself. In this sense, water is not only the solvent of life, but also its memory, carrying within it the history of all who have lived before. As this gentle reflection on water comes to a close, it is worth holding on to the simple truth that life and water are inseparable. Every thought you have, every step you take, every quiet moment of rest depends on this clear, unassuming liquid. In its stillness and in its motion, it supports you, surrounds you, and sustains you. Biochemistry, for all its complexity, rests on this foundation. And so, in learning about water, we are not only studying chemistry, we are remembering the very essence of life itself. 6. Acids, bases, and pH. As we continue our calm journey into biochemistry, it is natural to pause and notice another subtle but essential aspect of the chemistry of life: the balance between acids and bases. In the same way that life depends on stable temperatures and gentle flows of water, it also depends on a quiet equilibrium between hydrogen ions and hydroxide ions. This balance is expressed through a scale we call pH, and though it may sound abstract, it is in fact a constant presence in every living system. The concept of pH is not merely a mathematical measure, but a way of understanding how life maintains harmony at the molecular level, ensuring that reactions proceed at the right speed, in the right place, and with the right outcome. The idea of acids and bases begins with water itself. Each water molecule is capable of splitting into two charged parts: a hydrogen ion, which is essentially a single proton, and a hydroxide ion, which is the remainder of the molecule. Normally, water contains equal numbers of both, and so it is neutral. But if something increases the number of hydrogen ions, the solution becomes acidic. If something increases the number of hydroxide ions, the solution becomes basic or alkaline. The pH scale is simply a way of describing this balance, with values below 7 representing acidity, values above 7 representing alkalinity, and 7 itself standing as the calm midpoint of neutrality. Though this scale stretches from 0 to 14, most living systems operate within a narrow, gentle range, usually between 6 and 8, where the conditions are stable enough for the delicate machinery of cells. Acids are substances that release hydrogen ions when dissolved in water. They can be strong, releasing large numbers of ions at once, or weak, releasing them slowly and in smaller amounts. Bases, by contrast, are substances that accept hydrogen ions or release hydroxide ions, reducing acidity and creating a different type of chemical environment. In the body, acids and bases are not foreign or unusual. They are constant companions, appearing in digestion, in metabolism, in respiration, and in nearly every cellular process. The stomach, for instance, produces hydrochloric acid, lowering the pH to levels around 2, strong enough to break down food and kill harmful microbes. Blood, by contrast, maintains a much narrower range, close to 7.4, where enzymes can function properly and oxygen can be transported efficiently. This maintenance of pH is known as acid-base balance, and it is one of the most carefully guarded aspects of homeostasis. Even small deviations can have profound effects. If the blood becomes too acidic, a condition known as acidosis, the function of proteins and enzymes is disrupted, and the delivery of oxygen is impaired. If it becomes too alkaline, or alkalosis, the excitability of nerves and muscles increases, leading to instability. The body therefore invests great effort in keeping pH within its proper range, using several layers of defense. Buffers, such as the bicarbonate system in the blood, act as immediate stabilizers, soaking up excess hydrogen ions when they are too abundant, or releasing them when they are too scarce. The respiratory system contributes by controlling the release of carbon dioxide, which dissolves in water to form carbonic acid. By breathing faster or slower, the body can subtly adjust acidity. The kidneys provide a longer-term solution, excreting hydrogen ions in urine and conserving or releasing bicarbonate as needed. Together, these systems form a network of quiet guardians, ensuring that pH remains steady. It is worth noting that enzymes, the catalysts of life's chemistry, are exquisitely sensitive to pH. Each enzyme has an optimal range where it functions most efficiently, and outside of this range, its structure may shift, reducing its activity. For example, the enzyme pepsin in the stomach is adapted to work best in the highly acidic environment created by gastric acid. In contrast, enzymes in the small intestine require a more neutral to slightly basic environment, provided by secretions from the pancreas. This compartmentalization ensures that digestion proceeds smoothly, each step occurring in the environment that suits it best. The same principle applies throughout the body. The right pH creates the right conditions for life's reactions, like the tuning of an instrument that allows music to flow. Acids and bases also play roles in energy transfer. The gradient of hydrogen ions across membranes is one of the most important forces in biology, known as the proton gradient. In mitochondria, protons are pumped across the inner membrane, creating a difference in concentration. This gradient is then used to power the synthesis of ATP, the universal energy currency of the cell. Here, the controlled movement of hydrogen ions is not a byproduct but the very essence of energy conversion. Without the subtle management of acidity across membranes, cells could not produce the energy required to sustain life. It is another example of how the smallest details, invisible to the eye, shape the largest truths of existence. Beyond the body, acids and bases also shape the environments in which life is found. Oceans, for example, maintain a pH close to 8, slightly basic, and this stability is essential for marine organisms. When excess carbon dioxide dissolves in seawater, it lowers the pH, a process known as ocean acidification. This subtle shift affects corals, shellfish, and countless microorganisms, reminding us that pH balance is not only a cellular concern but a planetary one. Soil pH also determines which plants can thrive, influencing agriculture and ecosystems. Each organism, from the smallest bacterium to the largest forest, exists within a particular pH window, adapted to its environment, shaped by it, and contributing in return. What makes pH so fascinating in a gentle sense is its dual nature. On one hand, it is a simple measure of hydrogen ion concentration, a number on a scale. On the other, it is a profound reflection of balance, harmony, and the need for stability in a world that is always in flux. Life does not demand that conditions be perfect, only that they remain within a narrow, supportive range. And within that range, extraordinary complexity can unfold. This principle of balance is echoed throughout nature. Not too hot, not too cold, not too dry, not too wet. Always, it is balance that allows life to flourish. And pH is one of the quietest yet most essential examples of this truth. To reflect on acids, bases, and pH is to remember that life is delicate, yet resilient. A few ions, too many or too few, can shift the environment drastically. Yet systems exist to restore harmony, to bring the balance back. This constant adjustment, this quiet vigilance, is part of what defines living systems. They are not static, but always adapting, always maintaining conditions where their chemistry can continue. It is a reminder that harmony is not a fixed state but an ongoing process, like the gentle rocking of a boat that stays upright not by being rigid but by responding to each wave. As this exploration of acids, bases, and pH draws to a close, it becomes clear that this aspect of biochemistry, though subtle, is deeply significant. The scale of pH may be measured in numbers, but its meaning is felt in every breath, every heartbeat, and every reaction that sustains life. It is the quiet guardian of balance, the unseen thread that holds together the fabric of metabolism, energy, and structure. In the calm language of chemistry, pH is not only a measure of acidity, but also a measure of life's commitment to balance, its ability to endure through constant adjustment, and its gentle insistence that harmony is the path to survival. 7. Organic chemistry of life. When we begin to explore the chemistry of life in more detail, we naturally find ourselves drawn into the study of organic chemistry. This branch of chemistry is devoted to carbon and the endless shapes it can create. Organic chemistry may seem at first like an abstract science filled with formulas and structures, but in truth, it is the quiet foundation of every living system. To speak of organic chemistry is to speak of the forms that carbon, hydrogen, oxygen, nitrogen, and a few other elements can weave together, building the molecules that sustain cells, carry energy, and pass information from one generation to the next. It is here that the architecture of life takes shape, and though the structures may appear small, they are the patterns from which all complexity emerges. The defining feature of organic chemistry is carbon's unique ability to form 4 covalent bonds. This simple fact opens a universe of possibilities. Carbon can bond with other carbon atoms, creating long chains that twist and branch. It can form rings that provide stability and symmetry. It can bind with hydrogen to make hydrocarbons, with oxygen to form alcohols and acids, with nitrogen to form amines, and with sulfur or phosphorus to create even more variation. Each combination gives rise to a different type of molecule, with properties shaped by the functional groups that hang from the carbon skeleton. These functional groups, hydroxyl, carbonyl, amino, carboxyl, phosphate, and sulfhydryl, are like the accents and inflections in a language, changing the meaning of the same root word into something new. Take hydrocarbons as a starting point. These simple molecules, made only of carbon and hydrogen, are the scaffolds upon which more complex structures are built. In the body, hydrocarbon chains form the tails of fatty acids, creating lipids that store energy and form membranes. Their nonpolar nature means they do not mix well with water, and this property is not a flaw, but a gift. By avoiding water, hydrocarbons allow cells to create barriers, membranes that separate the inside from the outside. In this way, the simplest of carbon structures contributes directly to one of life's most fundamental requirements, the ability to define a boundary, to have an inside that is distinct from the world beyond. Functional groups add variety and function. A hydroxyl group, consisting of an oxygen bonded to a hydrogen, makes molecules polar and soluble in water, as in alcohols and sugars. A carbonyl group, with a carbon double bonded to oxygen, introduces reactivity, forming aldehydes and ketones that participate in metabolic pathways. Amino groups, with their nitrogen and 2 hydrogens, make molecules basic and are central to the structure of amino acids. Carboxyl groups, combining carbon, oxygen, and hydroxyl, act as acids, releasing hydrogen ions and giving amino acids and fatty acids their character. Phosphate groups, rich in negative charge, carry energy in molecules like ATP and form the backbone of nucleic acids. Sulfhydryl groups, containing sulfur and hydrogen, allow the formation of disulfide bridges, stabilizing proteins in their folded forms. Each of these groups is small, yet when attached to a carbon skeleton, they transform its behavior entirely, creating the diversity needed for life's chemistry. Organic chemistry is also about isomerism, the ability of molecules with the same formula to exist in different arrangements. Structural isomers differ in the placement of their bonds, while geometric isomers differ in spatial orientation, and enantiomers are mirror images of each other, like left and right hands. Though these differences may seem subtle, they have profound consequences in biology. One enantiomer of a molecule may be biologically active, while its mirror image is not. Life is sensitive to these details, selecting one form and excluding the other, a reminder of how finely tuned biochemistry truly is. The molecules that arise from organic chemistry are not ends in themselves, but the components of larger systems. Sugars, with their carbon, hydrogen, and oxygen in precise ratios, serve as immediate sources of energy and as building blocks for more complex carbohydrates. Lipids, with their hydrocarbon tails and functional head groups, create membranes and store energy in concentrated forms. Proteins, built from amino acids, fold into countless shapes to serve as enzymes, receptors, and structural elements. Nucleic acids, with their sugar phosphate backbones and nitrogenous bases, carry the instructions for life. All of these molecules are variations on carbon's theme, made possible by the flexibility of organic chemistry. What makes this area of biochemistry so peaceful to reflect upon is the way it reveals continuity. The molecules in your body are not unique inventions. They are variations on forms that exist in all living things. Glucose, the simple sugar that fuels your cells, is the same glucose that fuels a plant, a fish, or a bacterium. The amino acids in your muscles are the same ones in the wings of a bird or the roots of a tree. The nucleotides in your DNA are the same as those in nearly every other organism on Earth. Organic chemistry provides a common language, spoken fluently by all of life, regardless of shape, size, or species. There is also an elegance in the way organic molecules are recycled. In the environment, carbon cycles between the atmosphere, organisms, soil, and oceans. Plants fix carbon from the air into sugars. Animals consume plants and use those sugars for energy. And microbes return carbon back to the earth and air through respiration and decomposition. The same carbon atoms are used again and again, sometimes as part of a leaf, sometimes as part of an insect, sometimes as part of your own body. Organic chemistry is not a static arrangement, but a flowing story, a cycle that has continued unbroken for billions of years. Life's reliance on organic chemistry also connects it to the broader cosmos. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are not unique to Earth. They are forged in stars and spread across galaxies. Meteorites contain amino acids, sugars, and other organic molecules, suggesting that the chemistry of life is not confined to our planet, but is a universal potential. When we look out into the night sky and wonder if life exists elsewhere, what we are really asking is whether organic chemistry has unfolded beyond Earth in the same gentle patterns. The universality of these molecules gives us reason to think it may have. As we reflect on organic chemistry, it becomes clear that what may appear complex is rooted in simple principles. Carbon bonds flexibly, functional groups add character, isomers create variation, and together they form the molecules of life. The combinations are endless, but the rules are few, and from this simplicity emerges the diversity of every living system. It is a reminder that complexity does not need chaos to arise. It can be built quietly, patiently, from the repetition of simple patterns. As this part draws to a close, imagine the molecules within you, sugars flowing in your blood, lipids forming the walls of your cells, proteins carrying out their tasks, and nucleic acids storing your genetic story. Each of them is a creation of organic chemistry, a pattern of carbon and its companions, arranged into forms that sustain life. Though they are too small to see, they are always present, always at work, and always connected to the greater cycles of nature. Organic chemistry is not just a subject for textbooks. It is the poetry of life, written in atoms, a quiet song that plays within every organism, every moment, without end. 8. Carbohydrates, sugars of energy. Carbohydrates are often thought of simply as sugars or starches, familiar to us as part of our daily meals. But in the calm world of biochemistry, they reveal themselves as far more than food. They are among the most ancient and universal molecules of life, found in every living organism, serving as a primary source of energy, as structural components, and even as signals that allow cells to recognize and communicate with one another. They are called carbohydrates because they are built from carbon, hydrogen, and oxygen, usually in a simple ratio where each carbon is paired with a water molecule. This arrangement gives them both diversity and harmony, a balance that makes them indispensable to the chemistry of life. At their simplest, carbohydrates exist as monosaccharides, single sugar molecules. Glucose is the most familiar, a 6-carbon sugar that flows gently through the bloodstream, carrying energy to every cell of the body. Its structure is not rigid but can exist in linear form or as a ring, with the ring being the most stable and most common in living systems. Other monosaccharides include fructose, found in fruits, and galactose, often linked with glucose to form lactose, the sugar in milk. Despite their small size, these molecules carry immense significance. They are the currency of energy, easily transported and readily broken down to release the fuel that cells need. When you breathe in oxygen and your cells harvest energy, it is usually glucose that is being oxidized, step by step, into carbon dioxide and water, releasing adenosine triphosphate, or ATP, along the way. Monosaccharides can join together to form disaccharides, 2 sugar molecules. Sucrose, the common table sugar, is made of glucose and fructose linked together. Lactose, the sugar in milk, is made of glucose and galactose. Maltose, formed from 2 glucose units, is a product of starch digestion. These disaccharides show how the same basic building blocks can combine into new forms with different properties. The bonds that link them, known as glycosidic bonds, are formed through dehydration reactions, in which a water molecule is released as the sugars join. These bonds are not permanent. They can be broken again through hydrolysis, with water re-entering to separate the sugars. The cycle of joining and breaking is part of the gentle rhythm of metabolism, flexible and reversible, allowing carbohydrates to move between storage and release as needed. Beyond these small sugars lie the polysaccharides, long chains of monosaccharides joined together. Starch, the storage carbohydrate of plants, is one of the most well-known. It is composed of glucose units linked in a way that allows the molecule to coil and branch, storing energy compactly, but making it accessible when needed. When humans and animals eat plants, enzymes break down starch into glucose, providing a steady supply of fuel. Glycogen serves a similar role in animals, stored primarily in the liver and muscles. It is highly branched, allowing glucose to be released quickly in times of demand, such as during exercise or fasting. In this way, glycogen is like a quiet reservoir, always ready to provide energy when the need arises. Carbohydrates also play structural roles. In plants, cellulose forms the rigid walls that give strength to stems, leaves, and wood. Cellulose is made of glucose, just like starch and glycogen, but the glucose molecules are linked in a different orientation, creating straight chains that bundle together into strong fibers. Most animals, including humans, cannot break down cellulose because we lack the necessary enzymes, yet it remains important in our diet as fiber, aiding digestion by providing bulk and supporting the movement of material through the intestines. In fungi and in the exoskeletons of insects and crustaceans, a different structural carbohydrate, chitin, appears. It too is a chain of glucose-like molecules, modified slightly to add nitrogen-containing groups. Strong and flexible, It is another example of how carbohydrates extend beyond energy storage into the architecture of life itself. On a smaller scale, carbohydrates are found decorating the surfaces of cells, attached to proteins and lipids. These carbohydrate chains, often complex and branching, act as markers of identity. They allow cells to recognize one another, to distinguish self from non-self, and to participate in communication. The ABO blood group system, for example, is determined by the specific carbohydrates present on the surface of red blood cells. This signaling role of carbohydrates is quiet but profound, reminding us that sugars are not only energy carriers but also conveyors of information, essential to the social life of cells. The metabolism of carbohydrates is one of the central themes of biochemistry. Glucose is broken down through glycolysis, a pathway that converts it into pyruvate while producing a small but essential amount of ATP. The pyruvate can then enter the citric acid cycle, where more energy is released. And finally, oxidative phosphorylation, where the majority of ATP is generated. This cascade of reactions, though complex, is steady and continuous, like a river flowing from its source to the sea. Each step is guided by enzymes, each reaction carefully regulated, ensuring that energy is released, neither too quickly nor too slowly, but in the measured rhythm that cells require. Carbohydrates also play a role in photosynthesis, the process by which plants, algae, and some bacteria capture sunlight and use it to build sugars from carbon dioxide and water. Here, the cycle runs in reverse. Energy is not released but stored, as carbon atoms are fixed into glucose and other sugars. These sugars then serve not only the plant itself but also the countless organisms that depend on plants for food. In this way, carbohydrates form the link between sunlight and the living world, capturing light energy and transforming it into a form that can flow through ecosystems. There is a quiet beauty in the way carbohydrates illustrate the principle of simplicity, giving rise to complexity. Glucose, a small molecule with just 6 carbons, can be arranged into starch, glycogen, cellulose, or chitin, each with entirely different properties and roles. The same sugar can fuel a cell, build a wall, or signal a neighbor, depending on how it is arranged and connected. Life does not require an endless variety of elements to achieve its diversity. It relies instead on a small set of building blocks that can be combined in infinite ways. Carbohydrates are one of the clearest examples of this truth. To reflect on carbohydrates is to see them not just as food or fuel, but as central actors in the drama of life. They are present in the bread that sustains us, in the leaves of trees reaching for the sun, in the shells of crabs, and in the fibers of cotton. They flow in our bloodstreams as glucose, ready to provide energy to every cell, and they stand firm in the trunks of forests as cellulose, providing strength and structure. They carry energy, they shape form, they share information, and they do so quietly, continuously, without fanfare. As this exploration of carbohydrates comes to a gentle close, one can appreciate them as more than simple sugars. They are the silent providers of energy, the architects of strength, the messengers of identity. Every time you take a breath, every time you move a muscle, carbohydrates are there, releasing energy to support you. Every time you see a tree or a field of grass, carbohydrates are there, giving shape and stability. They are the sugars of energy, the fibers of life, the quiet threads that weave together the fabric of living systems. In their simplicity lies their beauty, and in their universality lies their power, connecting all organisms through the shared chemistry of carbon, hydrogen, and oxygen, arranged in endlessly meaningful ways. 9. Lipids, silent energy stores. If carbohydrates are the immediate and flowing sources of energy, lipids are their quieter, longer-lasting companions. Lipids are a diverse group of molecules united not by a single structure but by a common property. They do not mix easily with water. This hydrophobic nature gives lipids a special role in life's chemistry. Where water creates fluidity and openness, lipids create boundaries, reserves, and insulation. They are the molecules of stillness, of storage, and of protection, quietly holding energy in concentrated form and shaping the membranes that enclose every living cell. To reflect on lipids is to reflect on the silent strength that sustains life. Not always visible, but always essential. Lipids come in many forms, but one of the simplest and most familiar are fats, also called triglycerides. Each fat molecule is composed of a glycerol backbone, a small 3-carbon molecule, joined to 3 fatty acids. Fatty acids themselves are long chains of carbon and hydrogen, sometimes with single bonds throughout, sometimes with one or more double bonds that introduce bends into the chain. Saturated fatty acids, with no double bonds, are straight and can pack tightly together, forming solid fats at room temperature. Unsaturated fatty acids, with double bonds, have kinks that prevent close packing, leading to oils that remain liquid. This difference in structure leads to differences in behavior, but both forms are united in their role as energy stores. A gram of fat contains more than twice the energy of a gram of carbohydrate, making lipids the most efficient way for organisms to save energy for future needs. When food is scarce, when winter arrives, when migration requires long journeys, it is the energy stored in fats that sustains life. Beyond simple fats, lipids take on other forms. Phospholipids, for example, are molecules where one fatty acid in a triglyceride is replaced by a phosphate group. This small change creates a profound shift in behavior. The fatty acid tails remain hydrophobic, avoiding water, but the phosphate head is hydrophilic, attracted to water. As a result, phospholipids arrange themselves spontaneously into bilayers, with the tails facing inward and the heads facing outward. This arrangement forms the basis of cell membranes, the barriers that separate the inside of the cell from its environment. These membranes are not rigid walls but flexible, dynamic structures, allowing the cell to maintain its identity while still interacting with the world around it. The dual nature of phospholipids, part water-loving, part water-avoiding, is the quiet trick that makes life possible. Steroids are another class of lipids, built not from long chains but from 4 fused carbon rings. Cholesterol is the most familiar steroid, often misunderstood in popular conversation, but indispensable in biology. It helps maintain the fluidity of cell membranes, keeping them from becoming too rigid or too loose. Cholesterol also serves as the precursor for steroid hormones, such as estrogen, testosterone, and cortisol. These hormones act as chemical messengers, guiding development, metabolism, and stress responses. Though small in quantity compared to fats or phospholipids, steroids carry influence far greater than their numbers suggest, shaping the physiology of entire organisms with quiet precision. Lipids also appear as waxes, long-chain molecules that form protective coatings. Plants coat their leaves with waxes to prevent water loss, allowing them to thrive in dry environments. Animals use waxes in fur and feathers for waterproofing, enabling survival in rain or aquatic habitats. Bees produce wax to build honeycombs, perfect structures that store food and shelter their young. These uses may seem simple, but they are vital, another way in which lipids act as guardians of stability and endurance. In energy metabolism, lipids provide more than storage. When fatty acids are broken down, they enter pathways that generate large amounts of ATP. This process, known as beta oxidation, takes place in the mitochondria, where carbon units are removed 2 at a time and converted into acetyl-CoA, which then feeds into the citric acid cycle. The result is a slow, steady release of energy, far greater than what carbohydrates can provide, though requiring more time. This distinction reflects the complementary roles of carbohydrates and lipids. Carbohydrates offer immediate fuel, while lipids hold long-term reserves. In times of abundance, energy is stored as fat. In times of scarcity, fat becomes the lifeline that allows survival. Lipids also play roles in signaling beyond hormones. Certain lipid molecules act as messengers within cells, part of signaling pathways that regulate growth, inflammation, and immune responses. These signals are not loud or chaotic but subtle, precise instructions that guide cellular behavior. The dual role of lipids as both structural components and messengers highlights their versatility. They are not only passive stores of energy, but also active participants in the orchestration of life. It is worth reflecting on the quiet presence of lipids in daily life. In the human body, fat provides cushioning for organs, insulation against cold, and reserves of energy. In animals, fat layers allow hibernation through long winters or fuel migration across vast distances. In seeds, lipids provide the stored energy that allows a plant to sprout and grow before it can make its own food through photosynthesis. Everywhere in nature, lipids serve as silent guardians of continuity, ensuring that energy and protection are available when needed. Though lipids are often viewed with suspicion in modern diets, within the calm perspective of biochemistry, they are neither villains nor luxuries. They are fundamental. Without lipids, cell membranes could not exist. Without lipids, energy could not be stored in efficient form. Without lipids, hormones could not signal growth or reproduction. Life is not possible without them, and their presence in every organism speaks to their importance. They are not an excess, but an essence, a natural part of the balance that sustains living systems. There is a certain beauty in the way lipids contrast with water. While water flows, dissolves, and carries, lipids resist, hold, and protect. Together, they create the conditions for life. Water fills the cell, dissolving nutrients and carrying signals. Lipids enclose the cell, holding it together and allowing it to interact selectively with its surroundings. This partnership is not conflict but complementarity, two opposites working together to make harmony. Without water, reactions would halt. Without lipids, cells would have no shape. Together, they allow the quiet miracle of life to continue. As this gentle reflection on lipids comes to a close, one can see them as more than molecules of fat or oil. They are the silent energy stores, the builders of boundaries, the messengers of balance. They hold strength without noise, stability without rigidity, and energy without haste. Every living organism depends on lipids, from the smallest bacterium with its delicate membrane to the largest whale with its thick, insulating blubber. They are the quiet keepers of energy, the unseen architects of cells, and the guardians of survival. To understand lipids is to appreciate the silent work that sustains life. Always present, always steady, always essential. 10. Proteins. The workhorses of life. Among the many molecules that sustain life, proteins stand out as the most versatile and dynamic. If carbohydrates are the quick energy providers and lipids are the silent energy stores, proteins are the doers, the workers of the cell, shaping structure, catalyzing reactions, transporting molecules, sending signals, and defending against threats. They are so central that the word itself, protein, comes from a Greek root meaning of primary importance. In the gentle rhythm of biochemistry, proteins can be seen as tireless artisans, each folded into a precise shape that allows it to carry out a unique task. They are not only abundant but also endlessly varied, each one a quiet demonstration of life's creativity at the molecular level. Proteins are polymers, long chains made from smaller building blocks called amino acids. There are 20 standard amino acids used by living organisms, each sharing a common backbone of a central carbon atom, a hydrogen atom, an amino group, and a carboxyl group. What makes each amino acid unique is its side chain, or R group, which can be as simple as a single hydrogen, as in glycine, or as complex as a ring structure, as in tryptophan. These side chains give amino acids their character. Some are nonpolar and hydrophobic, preferring to avoid water, while others are polar or charged, interacting readily with water and other molecules. When linked together into chains through peptide bonds, amino acids form polypeptides, and when these chains fold into their final shapes, they become proteins. The folding of a protein is a process both intricate and natural. The sequence of amino acids, known as the primary structure, determines everything that follows. Hydrogen bonds between backbone atoms create local structures such as alpha helices and beta sheets. The secondary structure, interactions among side chains, then fold the entire polypeptide into a three-dimensional shape. The tertiary structure, in some cases, multiple polypeptides come together to form a larger complex. The quaternary structure, each level of organization is guided by the chemistry of bonds and interactions. From strong covalent disulfide bridges to gentle van der Waals forces, the final shape is not arbitrary. It is dictated by the sequence itself, a quiet code that ensures each protein takes the form most suited to its task. Proteins serve in countless roles. Structural proteins provide support and stability. Collagen, for instance, is the most abundant protein in the human body, forming strong fibers in connective tissues, skin, tendons, and bones. Keratin forms hair, nails, and the outer layers of skin, offering protection and resilience. These proteins act as the scaffolding of life, giving shape and strength to organisms. Enzymes, another class of proteins, act as catalysts, speeding up chemical reactions without being consumed. They lower the activation energy required for reactions, making processes that would otherwise be too slow proceed at the pace of life. Digestive enzymes like amylase and protease break down food into smaller molecules, while metabolic enzymes like hexokinase guide glucose into pathways that release energy. Each enzyme is highly specific, shaped to fit its substrate like a key to a lock. The quiet precision of enzymes is one of the most remarkable aspects of biochemistry, allowing life to unfold with speed and accuracy, yet always under control. Transport proteins carry molecules across barriers. Hemoglobin, found in red blood cells, binds oxygen in the lungs and delivers it to tissues throughout the body. Its structure allows it to pick up oxygen when concentrations are high and release it when they are low, a gentle responsiveness that sustains every breath. Membrane transport proteins form channels and pumps, moving ions and molecules across the lipid bilayer. Without them, cells would be unable to take in nutrients, expel wastes, or maintain the delicate balance of ions that underlies nerve impulses and muscle contractions. Proteins also serve as messengers. Hormones such as insulin are proteins that circulate in the blood, signaling cells to take up glucose and store energy. Receptor proteins, embedded in cell membranes, listen for these signals, binding hormones or neurotransmitters and triggering changes within the cell. This communication is subtle, like whispers carried across a crowded room, but it ensures that the body functions as a coordinated whole. In defense, proteins act as protectors. Antibodies, produced by the immune system, are proteins designed to recognize and neutralize foreign invaders such as bacteria and viruses. They bind specifically to their targets, marking them for destruction. Clotting proteins in the blood prevent excessive bleeding when injury occurs, forming nets that trap cells and seal wounds. These defensive roles show how proteins not only build and catalyze but also guard, maintaining the integrity of the organism. Proteins are also involved in movement, Actin and myosin, two structural proteins, interact in muscle fibers to create contraction, allowing organisms to move, lift, and breathe. Within cells, motor proteins like kinesin and dynein walk along microtubules, carrying vesicles and organelles from one place to another. These movements are not random but organized, guided by the precise interactions of proteins, like workers moving supplies through a well-planned city. One of the most remarkable aspects of proteins is their adaptability. The same 20 amino acids, arranged in different sequences, can form millions of unique proteins, each with a distinct shape and function. This versatility explains why proteins are often called the workhorses of life. They are not limited to one role or one environment but can be adapted to countless tasks. This adaptability also reflects the efficiency of life's design. From a limited set of building blocks comes an infinite variety of functions. Proteins also have life cycles. They are synthesized by ribosomes, folded into shape with the help of chaperone proteins, and eventually degraded when they are no longer needed. This constant turnover ensures that damaged or outdated proteins are removed and replaced, keeping the system fresh and functional. The recycling of amino acids mirrors the larger cycles of nature, where nothing is wasted and everything finds use again. Reflecting on proteins brings a sense of wonder at their quiet effectiveness. They are at work every moment, in every cell, unseen yet indispensable. Each heartbeat, each thought, each breath depends on proteins carrying out their tasks. They are the enzymes in your saliva, breaking down sugars as you eat. They are the hemoglobin in your blood, delivering oxygen as you read these words. They are the collagen in your skin, giving it strength, and the antibodies in your immune system, standing guard against harm. Without proteins, life would not simply be weaker, it would not exist at all. As we come to the end of this reflection, proteins reveal themselves as more than molecules. They are the artisans and guardians of life, each shaped by chemistry into a form perfectly suited to its purpose. They fold, they move, they build, they signal, they defend, they do not rest, yet they work without noise or complaint, silently carrying out the tasks that make life possible. To understand proteins is to glimpse the tireless dedication of nature at the molecular level, where structure and function merge into harmony. In their endless variety and constant labor, proteins remind us that life is not only sustained by energy and matter, but also by work, quiet, steady, and unceasing, the work that proteins perform in every living moment. 11. Enzymes, nature's catalysts. In the vast and intricate landscape of biochemistry, enzymes hold a special place of quiet power. They are proteins, yet their role is so profound and unique that they deserve their own reflection. Enzymes are catalysts, meaning they speed up chemical reactions without being consumed themselves. In the absence of enzymes, many of the reactions required for life would occur so slowly that they would be effectively useless. The gentle energy of the sun, the nutrients in food, the oxygen we breathe, none of these could be transformed into usable forms fast enough to sustain living systems without the steady work of enzymes. They are the subtle accelerators of life's chemistry, ensuring that every reaction happens at the right speed, in the right place, at the right time. To understand enzymes, one must first imagine what it means for a reaction to take place. In chemistry, molecules must collide with enough energy and in the correct orientation for their bonds to break and new ones to form. This required energy is called the activation energy. It is like a small hill that molecules must climb before they can roll down into a more stable arrangement. In the world outside of living systems, heat or pressure can provide this energy. But life is fragile, and cells cannot withstand extreme heat or harsh conditions. Enzymes solve this problem by lowering the height of the hill. They do not change the starting or ending points of a reaction, They simply make the journey smoother, faster, and more efficient. The way enzymes work is a quiet marvel of precision. Each enzyme has an active site, a region shaped in such a way that it fits a specific molecule, known as the substrate. The fit is often described as a lock and key, though it is more dynamic than this. When the substrate enters the active site, the enzyme undergoes a subtle change, adjusting itself to hold the substrate more snugly. This is called induced fit, and it ensures that the reaction proceeds with great efficiency. Once the substrate is bound, the enzyme stabilizes the transition state, lowers the activation energy, and facilitates the breaking and forming of bonds. The product of the reaction is then released, and the enzyme is free to catalyze the process again. In this way, a single enzyme molecule can assist in thousands or even millions of reactions, tirelessly repeating its role without wear. Enzymes are astonishingly specific. Each is designed for a particular reaction, or at least a narrow set of related reactions. This specificity ensures that the complex network of reactions within a cell does not dissolve into chaos. Instead, each pathway is guided, each step controlled. The enzyme, hexokinase for instance, catalyzes the first step in glycolysis, attaching a phosphate group to glucose. Without it, glucose could not be properly metabolized for energy. The enzyme DNA polymerase catalyzes the replication of DNA, ensuring that genetic information is faithfully copied. Without it, life could not reproduce. Each enzyme is like a skilled craftsman, designed for one task and performing it with quiet perfection. What is especially remarkable is how sensitive enzymes are to their environment. They require specific conditions of temperature and pH to function at their best. Too much heat can disrupt their structure, unfolding them and destroying their activity. Too much acidity or alkalinity can interfere with the charges that stabilize their shape. This sensitivity reflects the delicate balance of life, where conditions must remain within narrow ranges to support the function of enzymes. Yet within those ranges, enzymes operate with incredible efficiency, some increasing the rate of reactions by factors of millions or even billions. This efficiency allows cells to perform countless reactions simultaneously, all coordinated and all under control. Enzymes also play a role in regulation. Many are not always active but respond to signals, turning on or off as needed. Feedback inhibition is a common method, where the product of a pathway inhibits an enzyme at the beginning, preventing the cell from producing more than it needs. In this way, enzymes contribute to homeostasis, maintaining balance and preventing waste. Other enzymes are activated only when certain molecules bind to them or when they are modified by the addition of phosphate groups. These regulatory mechanisms allow cells to adapt, responding to changes in the environment with subtle shifts in their chemistry. Enzymes are not just catalysts but also guardians of balance, ensuring that life continues smoothly and efficiently. In the broader world of biology, enzymes shape processes that define life itself. In digestion, enzymes break down food into its simplest components. Amylases split starch into sugars. Lipases break fats into fatty acids. Proteases cut proteins into amino acids. Without these enzymes, nutrients would remain locked in large, inaccessible molecules. In photosynthesis, enzymes guide the steps that capture light energy and convert it into chemical energy stored in sugars. In respiration, enzymes orchestrate the breakdown of glucose and the production of ATP. Everywhere one looks in biochemistry, enzymes are at work, quietly facilitating the flow of energy and matter. Enzymes also have a hand in the way organisms interact with the environment. Plants produce enzymes that help them grow toward the sun and respond to changes in soil and weather. Microbes produce enzymes that allow them to digest unusual materials, from cellulose in wood to petroleum hydrocarbons in soil. In medicine, enzymes are used as treatments, such as enzymes that dissolve blood clots, or those that help with digestion in people with deficiencies. In industry, enzymes are employed in making cheese, brewing beer, washing clothes, and producing biofuels. Though they are tiny and invisible, their reach extends far beyond the cell, touching daily life in ways both subtle and profound. Reflecting on enzymes brings an appreciation for their balance of power and gentleness. They do not force reactions to occur that would otherwise be impossible. They simply allow what is possible to happen more quickly, more smoothly, and in the quiet conditions that life requires. They are not loud engines, but careful guides, lowering barriers and smoothing paths. Their work is unending, their presence constant, yet they remain unnoticed until studied closely. Every heartbeat, every breath, every thought relies on enzymes carrying out reactions in fractions of a second, and yet they do so without fanfare, without pause, without error. There is also a philosophical calm in the way enzymes embody cooperation. They do not act alone, but as part of networks and pathways, each step leading to the next. No enzyme is the whole story, but each contributes a piece, and together they sustain life. Their specificity ensures harmony, their regulation ensures balance, and their efficiency ensures survival. They are, in many ways, the quiet conductors of the symphony of life, unseen yet essential, ensuring that the music of biochemistry continues without interruption. As we close this reflection on enzymes, it is clear that they are more than catalysts. They are the quiet accelerators of existence, the guardians of speed and balance, the artisans of transformation. They take what is raw and make it usable. They guide molecules through transitions, and they do so endlessly, tirelessly, and with precision. Without them, life would not stumble, it would stop altogether. With them, it flows, steadily and calmly, through cycles of energy, growth, and renewal. To understand enzymes is to understand not only how life persists, but also how it persists with such grace, in harmony with the chemistry of the universe itself. 12. Nucleic acids, the story keepers. Among the great molecules of life, nucleic acids hold a role unlike any other. While carbohydrates provide energy, lipids create boundaries, and proteins perform countless tasks. Nucleic acids carry the instructions that guide all of these processes. They are the story keepers, the recorders of life's history, the molecules that ensure continuity from one generation to the next. Without them, there would be no inheritance, no memory written into the fabric of cells, no way for life to sustain itself across time. Nucleic acids are quiet scribes, writing, copying, and transmitting the codes that define existence, not with ink or parchment, but with sequences of chemical bases. There are two main types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. DNA is the long-term storage of genetic information, while RNA plays many roles, from carrying messages to forming part of the machinery that builds proteins. Both are polymers, long chains of smaller units called nucleotides. Each nucleotide is composed of three parts: a sugar, a phosphate group, and a nitrogenous base. The sugar is deoxyribose in DNA and ribose in RNA. The phosphate links one nucleotide to the next, creating a sugar-phosphate backbone that gives stability to the chain. The bases adenine, thymine, guanine, and cytosine in DNA, and adenine, uracil, guanine, and cytosine in RNA are the true bearers of information. It is the order of these bases, like letters in a language, that encodes the instructions for life. The structure of DNA is one of the most iconic discoveries in science. It forms a double helix, two strands twisted around each other, held together by hydrogen bonds between complementary bases. Adenine pairs with thymine, and guanine pairs with cytosine, always in the same way, creating a gentle regularity that ensures faithful copying. This complementarity is the secret of replication. When a cell divides, the two strands of DNA separate, and each serves as a template for a new strand. The result is two identical copies, one for each daughter cell. This simple, elegant mechanism allows information to be passed along with remarkable accuracy, generation after generation. RNA, though similar in composition, is more versatile and dynamic. Unlike DNA, which usually exists as a stable double helix, RNA is single-stranded and capable of folding into many shapes. Messenger RNA carries instructions from DNA to the ribosomes, where proteins are built. Transfer RNA brings amino acids to the ribosome, matching them to the appropriate sequence of the messenger. Ribosomal RNA forms the core of the ribosome itself, catalyzing the formation of peptide bonds. Beyond these roles, RNA molecules can regulate gene expression, catalyze reactions, and even serve as carriers of information in certain viruses. In this way, RNA shows us that nucleic acids are not passive record keepers but active participants in life's processes. The universality of nucleic acids is striking. Every organism, from the smallest bacterium to the largest whale, uses DNA and RNA in the same basic way. The genetic code, the set of rules by which sequences of bases are translated into amino acids, is nearly universal across all of life. This means that a gene from one species can often function in another, a fact that has been confirmed in countless experiments. This shared code is one of the strongest pieces of evidence for the common ancestry of all life. It also reflects a deep simplicity. Life has chosen one system to record its story, and that system has been preserved for billions of years. The information stored in nucleic acids is immense, but it is not just the quantity that matters. It is the precision of the sequence. A single change in one base, a mutation, can alter a protein, disrupt a pathway, or create a new function. Mutations are the source of both disease and evolution, reminders that the genetic code is both fragile and flexible. Some mutations are harmful, others neutral, and some beneficial, providing the raw material for adaptation and diversity. In this way, nucleic acids are not just record keepers but also storytellers, allowing life's tale to change, to branch, and to explore new directions. Nucleic acids also highlight the interplay between stability and change. DNA, with its double helix and complementary base pairing, provides stability, ensuring that information is preserved with high fidelity. RNA, with its single strands and varied roles, provides flexibility, allowing information to be expressed, regulated, and adapted. Together, they embody the balance that life requires, the permanence of memory, and the adaptability of expression. Without DNA, there would be no long-term inheritance. Without RNA, there would be no way to use that inheritance in the dynamic present. The two molecules complement one another, like past and present, continuity and change. It is worth reflecting on the scale of nucleic acids. A single human cell contains about 2 meters of DNA, tightly coiled and packed into chromosomes, yet invisible to the eye. Across the trillions of cells in the body, the DNA would stretch to the sun and back many times. And yet, this vast amount of material is composed of only 4 bases, repeated in different sequences. From this simplicity comes extraordinary complexity. The instructions to build a heart, a brain, or a pair of lungs are all encoded in the same 4-letter alphabet. This is one of the quiet wonders of biochemistry. How so much can arise from so little. How richness can emerge from repetition. Nucleic acids are also part of the great cycles of life. The nucleotides that compose DNA and RNA are not static but constantly synthesized, broken down, and recycled. When cells die, their nucleic acids are degraded, and their components are reused by other cells. Even in this recycling, the continuity of information is preserved. New molecules carry old instructions, linking past and future in an unbroken chain. Life does not waste, and nucleic acids remind us of this principle, as their parts are continually renewed while their messages endure. The discovery and understanding of nucleic acids transformed biology. Before their role was known, heredity was a mystery, explained only in vague terms. Once the structure of DNA was revealed, and once the processes of replication, transcription, and translation were uncovered, the mystery became a story, one that could be read, copied, and even altered. Today, entire genomes can be sequenced, revealing the full set of instructions for an organism. Yet even with this knowledge, there remains a sense of wonder. To read the code is not to exhaust its meaning. The story of life, written in nucleic acids, is not only about information, but about the way that information gives rise to living beings, each unique, each part of the greater continuity of existence. As this reflection on nucleic acids draws to a close, it becomes clear why they are called the story keepers of life. They carry the instructions that define what an organism is and how it functions. They ensure that this information is passed from parent to child, from generation to generation, linking the present to the past and the future. They allow for stability, yet they also allow for change, creating the possibility of growth, adaptation, and evolution. They are at once simple and profound. Small molecules, with the power to carry the story of life itself. To understand nucleic acids is to recognize that every living thing carries within it a written memory, not in words but in sequences of bases, a story told in the language of chemistry, unfolding quietly and continuously across the history of the Earth. 13. Energy in biochemistry. At the heart of every living system lies the gentle flow of energy. Life is not possible without it, for energy is the force that allows molecules to move, reactions to proceed, and cells to maintain their delicate order. Unlike the stillness of non-living matter, living systems are in constant flux, quietly balancing inputs and outputs, transforming one form of energy into another, always with a steady rhythm that sustains existence. In biochemistry, energy is not imagined as sudden explosions or violent surges, but as the calm, continuous current that carries life forward. To understand energy in biochemistry is to see how living things capture, store, and release it in ways that are both subtle and profoundly efficient. Energy itself takes many forms, but in the context of life, this is the energy stored in bonds between atoms, the quiet potential that can be released when bonds are broken and new ones are formed. Food, sunlight, and oxygen are not valuable in themselves, but because they carry energy that can be transferred into forms that cells can use. Biochemistry provides the pathways for these transfers, ensuring that energy is neither wasted nor spent too quickly, but harnessed in a measured and steady flow. Just as a river is guided by its banks, so too is energy in living systems, guided by enzymes and molecules that direct its path. One of the most important concepts in biochemistry is free energy, often described as the portion of energy available to do work. In a reaction, molecules move from higher energy states to lower energy states, releasing free energy in the process. This energy can then be captured and used to power other reactions, such as the building of proteins, the pumping of ions, or the movement of muscles. Reactions that release energy are called exergonic, while those that require energy input are called endergonic. Life is built on the coupling of these two kinds of reactions. Energy released from one process is used to drive another, creating a seamless web where nothing is wasted. It is a quiet efficiency, a constant balance between giving and receiving, release and capture. The most familiar carrier of energy in cells is adenosine triphosphate, or ATP. Often described as the energy currency of life, ATP is a small molecule with a big role. It consists of adenine, ribose, and 3 phosphate groups. The bonds between the phosphate groups are rich in potential energy, and when one phosphate is removed, ATP becomes ADP, releasing energy that can be used immediately for cellular work. This energy might drive the contraction of a muscle fiber, the transport of an ion across a membrane, or the synthesis of a new molecule. ATP is not stored in large quantities. Instead, it is continually regenerated, like a battery that is constantly recharged as it is used. In this way, ATP provides a steady, ongoing source of power, always available, always ready to be used. The generation of ATP is itself a remarkable story and of biochemical pathways. In glycolysis, glucose is broken down into smaller molecules, releasing a modest amount of energy directly and producing intermediates that feed into deeper cycles. The citric acid cycle, also known as the Krebs cycle, follows, breaking down molecules further and releasing high-energy electrons. These electrons are carried by molecules such as NADH and FADH to the electron transport chain, a series of protein complexes in the mitochondrial membrane. Here, electrons move step by step, each transfer releasing energy that is used to pump protons across the membrane, creating a gradient. This gradient, a quiet imbalance of charges, is then harnessed by the enzyme ATP synthase, which allows protons to flow back across the membrane, using their movement to generate ATP. It is a process both intricate and elegant, a molecular turbine powered not by fire or force, but by the steady movement of ions across a barrier. Energy in biochemistry is not only about release, but also about capture. In plants, algae, and certain bacteria, sunlight provides the input, and photosynthesis transforms this light energy into chemical energy stored in sugars. Pigments like chlorophyll absorb photons, raising electrons to higher energy states. These excited electrons move through a chain similar to the one in mitochondria, creating a proton gradient that drives ATP production. At the same time, electrons are used to build molecules of NADPH, another energy carrier. Together, ATP and NADPH provide the power to fix carbon dioxide into glucose and other carbohydrates. In this way, the energy of sunlight is captured and stored, not in fleeting light but in stable chemical bonds that can sustain life long after the sun has set. Energy flow in biochemistry also highlights the principle of efficiency. While no process is perfectly efficient, life has evolved systems that minimize waste and make use of byproducts. Heat is inevitably released, but even this has a role, maintaining body temperature in warm-blooded animals. Carbon dioxide, a product of respiration, becomes the raw material for photosynthesis in plants, completing a cycle between organisms. Oxygen, released by photosynthesis, becomes the electron acceptor in respiration. These cycles illustrate how energy moves not only within a single organism but across entire ecosystems, connecting all life in a quiet exchange of inputs and outputs. It is important to recognize that energy in biochemistry is not a thing but a flow, not a substance but a movement. Molecules carry it, release it, and capture it, but it never remains in one place for long. Energy moves from the sun to plants, from plants to animals, from food to ATP, from ATP to cellular work. At each step, it changes form, but it does not vanish. This flow is the essence of life, the constant transformation that allows systems to remain far from equilibrium, ordered and alive. Without this flow, molecules would drift toward stillness, reactions would cease, and life would fade into inanimate matter. The calm truth about energy in biochemistry is that it is both ordinary and extraordinary. Ordinary because it follows the same physical laws that govern all matter and energy in the universe. Extraordinary because life has found ways to channel these laws into cycles and pathways that create complexity, resilience, and beauty. A single glucose molecule, oxidized step by step, yields enough energy to power countless cellular tasks. A single photon of sunlight, captured in chlorophyll, begins a chain of reactions that can feed entire ecosystems. These transformations are not dramatic explosions, but steady flows, quiet exchanges that happen millions of times every second within every cell. Reflecting on energy in biochemistry brings a sense of harmony. It shows us that life is not a defiance of physical law, but an exquisite expression of it. Energy is not hoarded, but passed along, not wasted, but repurposed, not chaotic, but guided. Each cell is a microcosm of this flow, a tiny system, capturing, storing, and releasing energy with calm precision. Each organism is part of a larger flow, connected to others through cycles of oxygen, carbon, and sunlight. At every level, energy is the gentle current that ties everything together, uniting the smallest reactions with the grandest ecosystems. As this reflection comes to a close, it is worth remembering that every heartbeat, every thought, every movement is an expression of biochemical energy. It is ATP releasing a phosphate. It is electrons flowing through a chain. It is protons moving across a membrane. These invisible processes sustain the visible reality of life. Energy in biochemistry is not just a topic of study, but the essence of vitality, the quiet current that has flowed since life first appeared and that will continue as long as life endures. In its presence, we find the rhythm of existence, steady and serene, always moving, always sustaining, always carrying life forward. 14. Glycolysis, the first pathway. When we speak of energy in biochemistry, we inevitably come to the pathways that transform food into usable fuel. Among these, glycolysis holds a special place, for it is both ancient and universal. Every cell, whether in a bacterium, a plant, or a human being, relies on glycolysis as its first step in breaking down glucose, the simple sugar that is one of life's most essential fuels. The word itself means splitting sugar, and this is precisely what happens. A single molecule of glucose, with its 6 carbons, is gently divided into 2 molecules of pyruvate, each with 3 carbons. Along the way, a modest but vital amount of energy is captured in the form of ATP and electron carriers. To reflect on glycolysis is to reflect on the first quiet spark of energy release, the beginning of cellular respiration, and the pathway that has endured unchanged for billions of years. Glycolysis takes place in the cytoplasm, the fluid interior of the cell. Unlike many other processes that require specialized organelles, glycolysis happens everywhere, in every cell type. This universality suggests its great age. Long before mitochondria existed, long before oxygen filled the atmosphere, early cells used glycolysis to harvest energy from the sugars available to them. Its persistence today is a living memory of those ancient beginnings, a thread connecting modern life with its earliest forms. It is simple enough to function in the most primitive cells, yet efficient enough to remain central in even the most complex organisms. The pathway itself consists of 10 steps, each catalyzed by a specific enzyme. Though it may be tempting to see these steps as mechanical, they unfold with the same calm rhythm as all of biochemistry, each reaction flowing into the next, guided by subtle changes in energy and structure. The process can be divided into 2 phases: an investment phase, where energy is spent, and a payoff phase, where energy is gained. In this balance, glycolysis reflects one of life's gentle truths. Sometimes, energy must be given before more can be received. In the investment phase, 2 molecules of ATP are used to prepare glucose for breakdown. First, glucose is phosphorylated, receiving a phosphate group from ATP, which makes it more reactive and traps it within the cell. Then it is rearranged and phosphorylated again, forming fructose 1,6-bisphosphate, a molecule primed for division. These steps may seem costly, as they consume energy, but they are necessary to set the stage. Life invests in the future, trusting that the payoff will come. When the 6-carbon molecule is split into two 3-carbon molecules, the pathway begins to reveal its purpose. The payoff phase begins with the conversion of these 3-carbon molecules into more stable forms, releasing energy in the process. Electrons are transferred to NAD, forming NADH, carriers that will later deliver high-energy electrons to the word electron transport chain. ATP is generated directly at 2 points in the pathway, a process known as substrate-level phosphorylation. By the end of glycolysis, the initial investment of 2 ATP molecules has yielded 4, for a net gain of 2 ATP per glucose. While this may seem modest compared to the wealth of energy produced later in cellular respiration, it is enough to sustain cells when oxygen is absent. And in ancient life, it was the main source of power. The final product of glycolysis is pyruvate, a small but versatile molecule. Pyruvate stands at a crossroads. In the presence of oxygen, it enters the mitochondria, where it is converted into acetyl-CoA and fed into the citric acid cycle, leading to far greater energy yields. In the absence of oxygen, it can undergo fermentation, being reduced to lactate in animals or converted into ethanol and carbon dioxide in yeast. These alternative pathways allow glycolysis to continue, regenerating NAD so that glucose can still be broken down and ATP can still be produced, even in the absence of oxygen. This flexibility is one of the reasons glycolysis has endured. It provides energy under a wide range of conditions, ensuring that life can persist in both abundance and scarcity. To view glycolysis in a gentle way is to see not just a series of chemical reactions, but a pattern of balance. The pathway invests before it gains. It splits one into two. It produces both immediate rewards and future possibilities. It is a cycle of preparation and release, of modest returns that build toward greater outcomes. Though simple compared to later processes like the citric acid cycle and oxidative phosphorylation, glycolysis is indispensable. Without it, glucose could not be tapped for its energy, and the great flows of respiration and photosynthesis would stall before they began. There is also a beauty in the universality of glycolysis, no matter where one looks in the cells of plants drawing energy from the sun, in the muscles of animals running across the land, in the microbes inhabiting soil or ocean, glycolysis is at work. The enzymes may vary slightly, the regulation may differ, but the core pathway is the same. It is a reminder that life, in all its diversity, shares a common foundation. Beneath the variety of forms and functions, there is a shared chemistry, a quiet agreement that glucose shall be split into pyruvate, and that this act will release energy to sustain life. The regulation of glycolysis is another example of life's careful balance. The pathway is controlled at key steps, especially those that require the investment of ATP. Enzymes such as hexokinase and phosphofructokinase respond to the levels of ATP and other molecules in the cell. When energy is abundant, glycolysis slows, conserving resources. When energy is scarce, glycolysis accelerates, providing fuel. This feedback ensures that the cell neither wastes energy nor starves, maintaining homeostasis with quiet precision. The regulation is not a command but a conversation, molecules signaling to one another, adjusting the flow in response to the needs of the cell. Glycolysis also plays a role beyond energy production. Its intermediates are used as starting points for other pathways, contributing to the synthesis of amino acids, nucleotides, and lipids. In this way, glycolysis is not only a source of energy, but also a source of building blocks. The products of the pathway are not confined to respiration, but spread out into the broader metabolism, supporting growth and repair. This versatility reinforces the central place of glycolysis in biochemistry. It is not a narrow path, but a crossroads, linking energy, structure, and synthesis. Reflecting on glycolysis invites us to appreciate its quiet resilience. It does not produce the grandeur of oxidative phosphorylation, with its torrents of ATP, nor the elegance of photosynthesis, with its capture of sunlight. Instead, a pathway that has never been abandoned in all the history of life. Its simplicity is its strength, and its universality is its legacy. Every cell, from the earliest forms of life to the most complex, continues to rely on glycolysis. It is a reminder that sometimes the oldest ways are the most enduring, and that life does not always need grandeur to persist. Sometimes it needs only a steady path, repeated again and again, quietly sustaining existence. As this part closes, glycolysis stands revealed as more than a pathway. It is a foundation, an ancient and universal rhythm of life. It takes what is simple and makes it useful. It provides energy when little else is available. It prepares the way for greater processes to follow. It is not dramatic, but it is indispensable. In every breath you take, in every meal you eat, glycolysis is at work, splitting sugars, releasing energy, and passing the story of life forward, as it has done since the dawn of biology. Quiet, steady, and eternal. It is the first step on the path of cellular energy, the gentle beginning of life's ongoing song. 15. The citric acid cycle, after glycolysis, has gently split glucose into two molecules of pyruvate. The story of energy in biochemistry deepens. Pyruvate, that modest 3-carbon molecule, is not the end of the journey, but a gateway to more profound transformations. In the presence of oxygen, pyruvate enters the mitochondrion, the organelle often described as the powerhouse of the cell. Here, it is converted into acetyl-CoA, a 2-carbon molecule attached to coenzyme A, which acts like a guide leading it into the next great pathway. The citric acid cycle, also known as the Krebs cycle or the tricarboxylic acid cycle. This cycle is central to metabolism, a circular sequence of reactions that extracts energy from acetyl groups, releasing carbon dioxide and capturing high-energy electrons in carrier molecules. To reflect on the citric acid cycle is to reflect on the elegance of a repeating loop, a steady rhythm in which molecules are transformed again and again, always returning to the beginning, always providing energy and building blocks for life. The cycle begins when acetyl-CoA enters and joins with a 4-carbon molecule called oxaloacetate. Together, they form citrate, a 6-carbon molecule that gives the cycle its name. From this first step, the cycle unfolds gradually, like a story being told in chapters. Citrate is rearranged, oxidized, and decarboxylated, losing carbons in the form of carbon dioxide. These carbons, once part of glucose, are now released back into the environment, to be used again by plants in photosynthesis, or to dissolve into the oceans and atmosphere. What remains are molecules enriched with electrons, carried by NADH and FADH, ready to deliver their cargo to the electron transport chain. Each turn of the cycle releases energy, not in a rush, but step by step, measured and controlled. Though the citric acid cycle is often described in terms of energy production, it is also a hub of metabolism, connecting countless pathways. The intermediates produced at each step are not confined to the cycle, but can be diverted to form amino acids, nucleotides, and lipids. In this way, the cycle does not only break down molecules, but also provides raw materials for building. It is both catabolic, breaking down fuels, and anabolic, supplying building blocks. This dual role reflects the balance of life itself, destruction and creation intertwined, energy released while structures are formed. The cycle does not simply consume but also provides, sustaining the continuous flow of matter and energy. The energy captured in the citric acid cycle comes primarily in the form of reduced electron carriers. Each turn produces 3 NADH molecules and 1 FADH, both of which carry high-energy electrons. These electrons will later be passed along the electron transport chain, ultimately driving the production of ATP. The cycle also produces a small amount of ATP directly, or more precisely, GTP, in some organisms, which can be easily converted to ATP. While this direct yield may seem modest, the true value of the cycle lies in the electrons it harvests, the invisible currency that fuels the later stages of respiration. It is striking to note that the citric acid cycle is not a straight line but a circle. Unlike glycolysis, which moves from glucose to pyruvate in a linear fashion, the citric acid cycle regenerates its starting molecule, oxaloacetate, at the end of each turn. This regeneration allows the cycle to continue indefinitely, as long as acetyl-CoA is available. The circular design gives stability and continuity, ensuring that the process does not simply end but repeats, each turn linking seamlessly to the next. In this way, the cycle mirrors the rhythms of nature itself, where seasons return, tides repeat, and life renews. The citric acid cycle is also tightly regulated, adapting to the needs of the cell. When energy is abundant, certain enzymes are inhibited, slowing the cycle to conserve resources. When energy is scarce, these same enzymes are activated, accelerating the flow to generate more NADH, FADH, and ATP. This regulation ensures that the cycle is not wasteful, but responsive, turning faster or slower as required. It is a quiet responsiveness, a constant tuning of pace that keeps metabolism in harmony with the needs of the organism. Another layer of beauty in the cycle lies in its universality. Every aerobic organism, from bacteria to humans, relies on the citric acid cycle. Though details may vary across species, the core reactions are conserved. This suggests that the cycle emerged early in the history of life and proved so effective that it has remained largely unchanged. Its persistence across billions of years is a testament to its efficiency and elegance. Just as glycolysis links all life through a shared pathway, so too does the citric acid cycle remind us of our common origins, the shared chemistry that sustains us all. In reflecting on the citric acid cycle, one can also appreciate its role in the greater cycles of the Earth. The carbon dioxide released in each turn does not vanish but becomes part of the atmosphere, available for plants and algae to capture once more in photosynthesis. In this way, the cellular cycle links to the planetary cycle, individual metabolism contributing to the global exchange of carbon. Each breath you exhale carries the product of this cycle, a quiet release of carbon dioxide that will one day be taken up again by a leaf, a tree, or a field of grass. The connection is profound. Your own biochemistry is part of the larger rhythm of life on Earth. The citric acid cycle also provides a lesson in efficiency. It extracts as much energy as possible from each molecule of acetyl-CoA, leaving little unused. The process is thorough, ensuring that nearly all of the potential energy stored in glucose is captured and made available for the cell. This efficiency is necessary, for life cannot afford to waste energy. At the same time, the cycle's intermediates ensure flexibility, allowing the pathway to feed into other processes when building blocks are needed. The cycle is not only efficient but adaptable, able to serve both energy production and biosynthesis. When viewed calmly, the citric acid cycle is less a set of chemical steps than a meditation on continuity. Molecules enter, are transformed, release their energy, and are replaced by new molecules, while the cycle itself endures. It is an endless turning, not unlike the slow rotation of planets or the flow of water in a stream. Life does not need to invent new processes at every moment. It relies on cycles, repeating patterns that sustain stability through time. The citric acid cycle is one such pattern, a quiet loop that sustains every organism that breathes oxygen. As this reflection concludes, one can see the citric acid cycle not only as a biochemical pathway, but as a symbol of life's persistence. It takes what enters, transforms it, releases what is no longer needed, and returns to its beginning, ready to repeat. It is steady, reliable, and enduring. In your own cells, as you rest and breathe, the cycle is turning thousands of times every second, unseen but essential, a quiet rhythm beneath your existence. To understand the citric acid cycle is to glimpse one of life's deepest harmonies, a circle of chemistry that has been turning since the earliest days of life, and that continues patiently and faithfully in every living cell today. 16. Oxidative phosphorylation. After the citric acid cycle has finished its steady turning, producing reduced electron carriers in the form of NADH and FADH, the stage is set for the final and most energy-rich step of cellular respiration. This step is known as oxidative phosphorylation, and it represents the culmination of glucose breakdown, the place where the majority of ATP is produced. It is here that the quiet work of glycolysis and the citric acid cycle finds its greatest reward, as electrons are guided through a series of protein complexes. Protons are moved across a membrane, and a delicate gradient is formed and then harnessed. To reflect on oxidative phosphorylation is to reflect on one of the most elegant processes in all of biochemistry, a process that resembles a flowing river of electrons and a rising tide of protons, both converging to generate the energy that sustains life. Oxidative phosphorylation takes place in the inner membrane of the mitochondrion, a structure folded into cristae that increase its surface area. Within this membrane, the electron transport chain resides, a sequence of protein complexes and mobile carriers that pass electrons from one to another. These electrons originate in the NADH and FADH, molecules that were produced earlier. Each carries a pair of high-energy electrons, released when nutrients such as glucose were oxidized. Now, those electrons are delivered to the chain, beginning a journey of stepwise transfers. At each stage, a little energy is released, not in a rush but in measured amounts, enough to move protons across the membrane from the mitochondrial matrix into the intermembrane space. This movement establishes an electrochemical gradient, a reservoir of potential energy known as the proton motive force. The electron transport chain consists of 4 main complexes. Complex I accepts electrons from NADH, transferring them to a molecule called ubiquinone, while pumping protons across the membrane. Complex II receives electrons from FADH, passing them along without pumping protons, but still contributing to the overall flow. Complex III passes electrons from ubiquinone to cytochrome C, again moving protons across the barrier. Finally, Complex IV transfers electrons to oxygen, the final electron acceptor, reducing it to water. Without oxygen, this chain cannot proceed, and the flow of electrons halts. Oxygen, then, is not only something we breathe, but the quiet recipient of electrons, the molecule that completes the process and allows energy production to continue. As the chain operates, protons accumulate on one side of the inner membrane, creating both a concentration difference and a charge difference. This imbalance is not wasteful but purposeful. It represents stored energy, like water behind a dam, ready to flow back down if given the chance. The structure that allows this flow is ATP synthase, a remarkable enzyme that spans the inner membrane. ATP synthase provides a channel for protons to move back into the matrix, down their gradient. As they pass through, the enzyme rotates, and this mechanical motion drives the synthesis of ATP from ADP and inorganic phosphate. Each turn of the turbine produces energy, captured in the bonds of ATP, ready to fuel countless cellular tasks. The numbers involved are striking. From one molecule of glucose, oxidative phosphorylation alone can produce around 30 molecules of ATP, compared to the 2 from glycolysis. and the small amount from the citric acid cycle. This makes it the major source of energy in aerobic organisms, the step that transforms the earlier modest gains into a wealth of usable power. It is no exaggeration to say that oxidative phosphorylation sustains most of the energy demands of life as we know it. Every thought, every movement, every heartbeat relies on the steady turning of ATP synthase and the quiet flow of protons across the mitochondrial membrane. The elegance of oxidative phosphorylation lies not only in its yield but also in its design. The electron transport chain does not waste energy but releases it gradually, each step capturing a portion to move protons. If electrons moved directly from NADH to oxygen in a single step, most of the energy would be lost as heat. Instead, the chain provides a series of small, manageable transfers, allowing the energy to be stored in the proton gradient. This controlled release reflects the patience of life, using energy not recklessly but with precision, ensuring that it is harnessed effectively. Oxidative phosphorylation also reflects the intimate relationship between energy and order. The creation of a gradient, a state of imbalance, is what allows work to be done. Protons move from high concentration to low, from one side of the membrane to the other, and in that movement lies the ability to generate ATP. Life thrives on such imbalances, carefully maintaining them and then allowing them to resolve in ways that release energy. It is a balance between separation and flow, between difference and unity, between holding and releasing. This principle is not confined to biochemistry but can be seen in many aspects of nature, where tension and resolution create the conditions for movement and growth. There is also a calm universality in oxidative phosphorylation. It is not unique to humans or animals, but present in all aerobic organisms. Plants, too, use this process in their mitochondria, even as they capture light energy in chloroplasts. Bacteria perform oxidative phosphorylation on their plasma membranes, proving that the process does not depend on mitochondria, but can be adapted to different structures. The universality of this pathway underscores its efficiency and importance. It is not a specialized feature, but a fundamental one, chosen early in evolution and preserved across billions of years. The role of oxygen in oxidative phosphorylation also offers a perspective on life's connection to the environment. Oxygen is more than just the gas we inhale. It is the final link in the chain of energy transfer, the molecule that accepts electrons and allows the entire process to continue. Without oxygen, the chain halts, the proton gradient dissipates, and ATP production ceases. This reliance ties our metabolism directly to the air around us, to the plants that release oxygen in photosynthesis, to the balance of gases in the atmosphere. Each breath is not only a supply of oxygen, but a continuation of the flow of energy, connecting the chemistry within us to the cycles of the earth. Reflecting on oxidative phosphorylation brings a sense of harmony. It is a process at once mechanical and chemical, involving turbines and gradients, flows and transfers, yet all carried out by molecules too small to see. It is constant, happening in every cell, every moment, without pause. Its efficiency is astounding, yet its action is quiet, invisible, and taken for granted. It is a reminder of how much of life happens beyond perception, how every movement we make is powered by countless unseen processes, working with perfect coordination. As this reflection comes to a close, oxidative phosphorylation reveals itself as more than just a pathway of energy production. It is a flowing river of electrons, a rising tide of protons, a turning wheel of ATP synthase, all working together in quiet synchrony. It is the culmination of cellular respiration, the final step that transforms the energy of food into the energy of life. It is steady, continuous, and universal, a process that has endured since the earliest days of oxygen on Earth, and that continues in every living cell today. To understand oxidative phosphorylation is to glimpse the quiet machinery of life's power, always turning, always flowing, always sustaining the gentle flame of existence. Photosynthesis. While oxidative phosphorylation captures the quiet power of oxygen in producing ATP, photosynthesis represents the beginning of that story, the place where energy from sunlight is captured and stored in the bonds of molecules. It is through photosynthesis that energy from the sun, a seemingly unreachable and distant star, is drawn gently into the chemistry of life. Plants, algae, and certain bacteria perform this transformation, taking light, carbon dioxide, and water and turning them into sugars and oxygen. In doing so, they provide the foundation of nearly every food chain on Earth. Photosynthesis is not only a process but a bridge between the physical and biological worlds, connecting the energy of sunlight to the flow of life in forests, oceans, and even in our own bodies. To reflect on photosynthesis is to reflect on the calm generosity of nature, where light becomes life and energy is stored for all living things. The story of photosynthesis begins with pigments, molecules capable of absorbing light. The most familiar of these is chlorophyll, the pigment that gives plants their green color. Chlorophyll absorbs light most strongly in the blue and red regions of the spectrum, reflecting green, which is why leaves appear the way they do. When a photon of light strikes chlorophyll, it excites an electron, raising it to a higher energy level. This simple act, light energizing an electron, is the first step in capturing solar energy. The electron does not remain excited for long, but is passed along to other molecules, beginning a chain of transfers, much like the electron transport chain in mitochondria. In this way, light is converted into chemical energy, step by step, through a process of controlled movement. Photosynthesis occurs in two stages: the light reactions and the Calvin cycle. The light reactions take place in the thylakoid membranes of the chloroplast, where pigment molecules are organized into clusters called photosystems. In photosystem 2, light energy excites electrons that are passed along an electron transport chain, pumping protons across the thylakoid membrane and creating a gradient. This gradient powers ATP synthase, just as it does in mitochondria, producing ATP. At the same time, water molecules are split to replace the lost electrons, releasing oxygen as a byproduct. This release is gentle yet profound, for it is the source of the oxygen we breathe. Every exhalation we make traces back to the splitting of water in photosynthesis, an unseen gift of plants and algae. The electrons continue their journey to photosystem I, where they are re-energized by another photon of light. From here, they are transferred to NADP, forming NADPH, another energy carrier. Together, ATP and NADPH provide the energy and reducing power needed for the next stage. The light reactions do not themselves produce sugar, but they create the tools with which sugars can be made. They are like the preparation of ingredients in a kitchen, gathering what is needed for the main dish to come. The Calvin cycle, also known as the dark reactions or the light-independent reactions, takes place in the stroma of the chloroplast, the fluid-filled space surrounding the thylakoids. Here, carbon dioxide from the atmosphere is fixed into organic molecules. The enzyme responsible for the first step is rubisco, one of the most abundant proteins on Earth. Rubisco attaches carbon dioxide to a 5-carbon sugar, ribulose bisphosphate, creating a 6-carbon molecule that quickly splits into 2, 3-carbon molecules. Through a series of reactions powered by ATP and NADPH, these molecules are transformed into glyceraldehyde, 3-phosphate, a simple sugar that can be used to build glucose and other carbohydrates. The cycle regenerates ribulose bisphosphate, allowing it to continue indefinitely as long as carbon dioxide and energy carriers are available. The Calvin cycle does not proceed quickly, nor does it produce large amounts of sugar in a single turn. But with countless chloroplasts working in every leaf, and countless leaves working in every forest or field, the process accumulates into vast quantities of carbohydrate. These sugars fuel the plant itself, providing energy for growth, repair, and reproduction. Yet they also serve as food for animals, fungi, and microbes, passing the captured sunlight through ecosystems in the form of energy-rich molecules. Thus, photosynthesis is not only a process of self-sustenance but of sharing, providing the basis for nearly all other forms of life. The oxygen released during photosynthesis has reshaped the planet. Billions of years ago, before photosynthetic organisms flourished, Earth's atmosphere contained little free oxygen. With the rise of cyanobacteria and later plants, oxygen began to accumulate, eventually allowing aerobic respiration to evolve. This change, sometimes called the Great Oxidation Event, transformed the chemistry of the Earth and opened the door for complex, energy-hungry organisms, including ourselves. The oxygen you inhale with each breath is a direct product of photosynthesis, a continual offering of plants and algae to the rest of life. In this sense, every breath is a reminder of our connection to the green world. Photosynthesis also illustrates the principle of cycles that runs through all of biochemistry. The carbon dioxide released in respiration returns to the atmosphere, where it is taken up by plants in photosynthesis. The sugars produced by plants are consumed by animals, fungi, and microbes, who break them down for energy. Oxygen produced in photosynthesis is consumed in respiration, completing the circle. These cycles are not wasteful but efficient, ensuring that matter is reused and energy flows smoothly. In this quiet reciprocity, photosynthesis and respiration mirror one another, two halves of a greater whole, balancing inputs and outputs across the living world. What is especially striking is that photosynthesis does not merely serve the needs of plants. It sustains entire ecosystems, and indeed the entire biosphere. From the grasses that cover plains to the trees that form forests, from the algae that drift in oceans to the mosses that cling to rocks, photosynthetic organisms are the producers, the base upon which all others depend. Herbivores feed on them. Carnivores feed on herbivores. Decomposers recycle what remains. And through it all, the sugars created from sunlight continue to flow. Photosynthesis is the quiet generosity of life, turning light into food and sharing it freely with all. In a larger sense, photosynthesis connects the Earth to the cosmos. Sunlight that traveled across millions of kilometers is absorbed in a leaf, exciting electrons and fueling chemistry. Energy born in the heart of a star becomes part of the structure of a tree, a grain of wheat, or the oxygen in your lungs. This connection is humbling, reminding us that life on Earth is not separate from the universe but part of its unfolding story. Photosynthesis is the thread that links the energy of the sun to the breath of a child, to the wing beat of a bird, to the ripple of waves across the sea. As this reflection draws to a close, photosynthesis stands revealed not just as a biochemical pathway, but as a profound expression of life's ability to capture and share energy. It is the gentle transformation of sunlight into sugar, of light into matter, of potential into reality. It provides oxygen for our breath, food for our bodies, and energy for our cells. It connects organisms to ecosystems, and ecosystems to the planet, weaving all into a single fabric sustained by the Sunday. Quiet, continuous, and universal, photosynthesis is the silent song of life, a melody that has played for billions of years and that continues every moment in the green of leaves, the shimmer of algae, and the unseen work of microbes. To understand photosynthesis is to glimpse the generosity of nature, where light becomes life, and life becomes a gift to all. 18. Cell membranes and transport. In the quiet world of biochemistry, boundaries are as important as flows. Life cannot exist without separation, without the gentle distinction between the inside of a cell and the world beyond it. Yet this separation cannot be absolute, for no cell could survive if it were completely sealed off from its environment. It must take in nutrients, release wastes, exchange signals, and maintain balance. The structure that makes this possible is the cell membrane, a delicate yet resilient barrier formed from lipids and proteins. The cell membrane does not simply divide. It organizes, protects, and communicates, allowing the cell to remain distinct while still being connected. To reflect on membranes and the transport they allow is to reflect on the balance between closure and openness, between stillness and movement, between individuality and interconnection. The cell membrane is built primarily from a bilayer of phospholipids. Each phospholipid has a hydrophilic head, which is drawn to water, and two hydrophobic tails, which avoid it. When placed in water, phospholipids spontaneously arrange themselves so that the heads face outward, touching water, while the tails face inward, shielded from it. This arrangement creates a bilayer, a sheet that is both flexible and stable. It is this bilayer that forms the foundation of every cell membrane. The beauty of this structure lies in its duality. It is fluid, allowing movement of lipids and proteins within it, yet it is also firm enough to act as a boundary. It is self-assembling, requiring no instruction, yet it serves as the framework for the complexity of life. Within this bilayer, proteins are embedded or attached, and these proteins provide the specificity and functionality of membranes. Some proteins span the entire membrane, forming channels or pumps that allow molecules to pass through. Others act as receptors, sensing signals from outside and transmitting them inside. Still others serve as anchors, attaching the membrane to the cytoskeleton or to other cells. Together, lipids and proteins create a mosaic, often described as the fluid mosaic model, where components move within the bilayer like pieces drifting gently in a sea. This fluidity ensures that the membrane is not rigid but adaptable, capable of changing shape, repairing itself, and adjusting to the needs of the cell. Transport across membranes occurs in several ways, reflecting the variety of needs cells must meet. Some substances move freely by diffusion, slipping between the lipid molecules if they are small and nonpolar, such as oxygen or carbon dioxide. This process requires no energy, only a concentration gradient, with molecules moving from where they are more concentrated to where they are less. It is a passive and natural flow, a quiet evening out of differences. Water, though polar, also moves across membranes through diffusion, a process called osmosis. When water moves to balance differences in solute concentration, it ensures that cells maintain their proper shape and volume, neither swelling too much nor shriveling away. Other substances require assistance to cross the membrane. Larger or charged molecules, such as glucose or ions, cannot pass freely through the lipid bilayer. For these, the cell uses transport proteins. Some proteins form channels that open to allow specific molecules through, while others act as carriers, changing shape to move their cargo across. These processes, known as facilitated diffusion, still rely on concentration gradients and do not require energy. They are a way of allowing necessary materials to pass through without disturbing the balance of the membrane. When substances must move against their concentration gradients, from areas of lower concentration to areas of higher concentration, active transport is required. This process consumes energy, usually in the form of ATP, to power pumps that push molecules across the membrane. The sodium-potassium pump is a classic example, maintaining the gradient of sodium and potassium ions essential for nerve impulses and muscle contractions. Though active transport is more costly than passive diffusion, it is necessary for maintaining the conditions that support life. It is a reminder that balance often requires effort, that homeostasis is not passive but an active process of regulation. In addition to individual molecules moving across, entire packets of material can enter or leave the cell through bulk transport. Endocytosis brings material in, with the membrane folding inward to form a vesicle that carries the cargo inside. Exocytosis does the opposite, sending vesicles to fuse with the membrane and release their contents outside. These processes allow cells to take in large molecules, remove waste, or secrete substances like hormones and neurotransmitters. They highlight the dynamic nature of the membrane, which is not merely a barrier but a living, active interface with the environment. The cell membrane also plays a role in communication. Carbohydrates attached to proteins and lipids on the outer surface serve as markers, allowing cells to recognize one another. These glycoproteins and glycolipids are part of the immune system's ability to distinguish self from non-self, part of tissue cells' ability to organize into coherent structures, and part of how cells interact within multicellular organisms. Receptors in the membrane detect hormones, neurotransmitters, or environmental signals, and they transmit these cues inward, initiating responses that adjust metabolism, growth, or movement. In this way, the membrane is not only a physical boundary but also a sensory and signaling hub, attuned to the needs of the cell and the world beyond. Membranes are also essential in compartmentalization within cells. Eukaryotic cells, with their internal organelles, rely on membranes to define spaces such as the nucleus, mitochondria, endoplasmic reticulum, and Golgi apparatus. Each membrane creates a unique environment, allowing specialized processes to occur without interference. Mitochondria generate energy, chloroplasts capture sunlight, lysosomes break down waste, all made possible by membranes that maintain distinct chemical conditions. This compartmentalization increases efficiency, allowing complex organisms to thrive. At the same time, it reflects a simple principle. Life depends on both separation and connection, on creating boundaries while allowing communication across them. When viewed gently, cell membranes embody a philosophy of balance. They enclose but do not isolate. They protect but also allow exchange. They maintain stability while remaining flexible. They are both barriers and bridges, ensuring that the cell can remain itself while being part of a larger environment. This duality is not a flaw but a feature, the very reason membranes are so effective. In their thinness lies their strength. In their fluidity lies their resilience. On a broader scale, membranes remind us of the unity of life. Every organism, from the simplest bacterium to the most complex animal, relies on membranes built from the same basic phospholipid bilayer. This shared architecture speaks of common origins and shared solutions. Despite the diversity of life, the fundamental design of the cell membrane has endured, unchanged in its essentials, for billions of years. It is a testament to the effectiveness of this structure, a design so simple and yet so profound that it remains at the foundation of biology. As this reflection concludes, the cell membrane reveals itself as more than a boundary. It is the quiet mediator of life's exchanges, the protector of the cell's integrity, the stage upon which communication and transport take place. Through diffusion and osmosis, through pumps and channels, through vesicles and receptors, the membrane ensures that the cell remains alive, balanced, and responsive. It is a living interface, always active, always adapting, always essential. To understand membranes and transport is to understand the delicate balance between inside and outside, between self and environment, between stability and change. It is to glimpse the silent guardianship of the cell, a guardianship that sustains every heartbeat, every breath, and every thought. 19. Biochemistry in everyday life. Though biochemistry often feels like a subject confined to laboratories, textbooks, or microscopic observations, it is in fact the constant rhythm beneath our daily existence. Every breath, every step, every moment of thought and rest is guided by the gentle unfolding of biochemical processes. The principles we have traced through molecules, pathways, and cycles are not abstract. They are present in the food we eat, the air we breathe, the emotions we feel, and the health we maintain. To see biochemistry in everyday life is to see the hidden harmony that sustains our bodies and connects us to the world around us. Food is perhaps the most direct way in which biochemistry touches our lives. When we eat bread, fruit, or rice, the carbohydrates within are broken down by enzymes into glucose, which fuels glycolysis and later respiration, producing ATP for our cells. When we enjoy cheese, meat, or beans, the proteins are digested into amino acids, which are then reassembled into the proteins that form our muscles, enzymes, and hormones. Fats, present in oils, nuts, or dairy, provide energy stores, insulation, and the building blocks for membranes. Even vitamins and minerals, though needed in small amounts, act as cofactors for enzymes, allowing reactions to proceed smoothly. Eating, then, is not only a matter of pleasure or necessity, it is the quiet replenishment of the molecules our bodies depend on, the intake of raw materials that are transformed into energy and structure through the language of biochemistry. Breathing is another daily act that is deeply biochemical. With each inhale, oxygen enters the lungs and dissolves into the blood, where it binds to hemoglobin. This oxygen is carried to tissues, where it accepts electrons at the end of the electron transport chain, allowing oxidative phosphorylation to continue. With each exhale, carbon dioxide, produced by the citric acid cycle, leaves the body, completing a cycle of exchange between ourselves and the environment. Breathing may feel effortless, but it is in fact the outward expression of inner biochemical flows. The rhythm of respiration reflects the constant need for oxygen to sustain ATP production, the constant release of carbon dioxide as a byproduct, and the deep connection between cellular life and the atmosphere of the earth. Movement, too, is a story of biochemistry. When a muscle contracts, it is the sliding of actin and myosin filaments, powered by ATP. Each contraction is fueled by the hydrolysis of ATP, releasing energy that allows proteins to shift and fibers to shorten. Whether we walk slowly or run quickly, whether we lift a weight or simply hold ourselves upright, biochemistry is there, supplying the energy and coordinating the movement. Even the fatigue that follows exertion has biochemical roots. As ATP is depleted, lactic acid accumulates, and glycogen reserves are consumed. Recovery comes through the replenishment of these molecules, as biochemistry restores balance. Every step on a path, every gesture of the hand, every beat of the heart is guided by unseen molecular processes, all carried out with precision and rhythm. Our senses are also deeply connected to biochemistry. Vision depends on photoreceptor proteins in the eye that change shape when struck by light, initiating electrical signals interpreted by the brain. Taste arises from receptors that bind sugars, salts, acids, or bitter compounds, each recognition a biochemical interaction between molecules on the tongue and substances in food. Smell comes from receptors in the nose that detect volatile molecules, turning chemical presence into sensory experience. Hearing depends on ion channels in hair cells of the ear, which respond to vibrations with changes in membrane potential. Even touch relies on proteins that respond to pressure, temperature, and pain. In every case, sensation arises from biochemistry, translating the outside world into signals the brain can understand. Emotions, too, have a biochemical dimension. Neurotransmitters such as dopamine, serotonin, and norepinephrine carry signals between neurons, shaping mood, motivation, and memory. Hormones such as adrenaline prepare the body for action, increasing heart rate and releasing glucose into the blood, while cortisol adjusts metabolism in times of stress. Endorphins, small peptides, act as natural pain relievers and produce feelings of calm and pleasure. Love, joy, anxiety, and sorrow all involve complex networks of biochemical signals, a reminder that even our most profound experiences are accompanied by quiet flows of molecules. To understand this does not diminish their depth. Rather, it shows the harmony between chemistry and feeling, between matter and mind. Health and medicine are perhaps where biochemistry is most visibly present in daily life. When we take medicines, they act at the biochemical level, binding to enzymes, receptors, or channels to alter pathways and restore balance. Antibiotics interfere with bacterial enzymes. Pain relievers reduce the production of signaling molecules, and vaccines train the immune system to recognize proteins from pathogens. Illnesses themselves often arise from biochemical imbalances, deficiencies of enzymes, disruptions of signaling pathways, or changes in genetic instructions. Nutrition, exercise, and rest all act as ways of supporting the body's biochemistry, providing resources and conditions for its processes to unfold smoothly. Healing, in many ways, is the restoration of biochemical harmony. Even the natural cycles we live within are biochemical. The feeling of sleepiness at night and alertness in the morning comes from circadian rhythms, guided by biochemical oscillations of hormones such as melatonin and cortisol. Hunger and fullness are regulated by signals such as ghrelin and leptin, telling us when to eat and when to stop. Growth, reproduction, and aging all reflect the long-term unfolding of biochemical programs written in DNA and expressed through proteins. We may not always notice these rhythms, but they shape the flow of our days and years, providing continuity and balance. On a larger scale, biochemistry links us to the ecosystems we inhabit. The oxygen we breathe is released by photosynthesis. The food we eat originates from carbohydrates and proteins built in plants or animals, and the carbon dioxide we exhale returns to the atmosphere to be used again. Decomposers in soil and water break down organic matter, recycling nutrients that sustain new growth. The cycles of carbon, nitrogen, and water all have biochemical dimensions, connecting the life of the smallest microbe to the life of the largest forest. To see biochemistry in everyday life is to see our place in these cycles, not as separate beings but as participants in a larger network of exchanges. Reflecting on these connections reveals that biochemistry is not distant or abstract but intimate and continuous. It is in the sugar, broken down in a morning meal, in the oxygen carried through the blood, in the contraction of muscles as we move, in the flow of neurotransmitters as we think and feel, in the repair of tissues as we heal, and in the rhythms of sleep as we rest. It is the language that cells speak quietly, the harmony that sustains both the smallest processes and the grandest experiences. Every part of daily life is touched by biochemistry, every act a living demonstration of molecules working together. As this reflection draws to a close, biochemistry stands revealed as the unseen companion of our lives. It is the gentle science behind eating and breathing, the foundation of movement and thought, the rhythm of health and healing. It is in the joy of a meal shared, the comfort of rest, the strength of activity, and the depth of emotion. To understand biochemistry in everyday life is to see that we are not separate from the molecular processes described in books, but embodiments of them, living expressions of the chemistry that sustains the earth. Quiet, steady, and always present, biochemistry is the melody beneath our days, the soft song of life itself. 20. Gentle conclusion. As we come to the end of this peaceful journey through the landscapes of biochemistry, it is natural to pause and reflect on the quiet truths we have encountered. We have walked slowly through the foundations of life's chemistry, from the elemental palette of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, to the bonds that hold atoms together, to the molecules that form the fabric of cells. We have explored the sugars that provide energy, the lipids that store and protect, the proteins that work tirelessly, and the nucleic acids that carry the story of existence across generations. We have followed the flow of energy through glycolysis, the citric acid cycle, oxidative phosphorylation, and photosynthesis, and we have seen how membranes, enzymes, and cycles sustain life's delicate balance. Each part has been a gentle step in understanding that biochemistry is not only a science but also a quiet narrative of how life persists, adapts, and endures. What emerges from this journey is the realization that life is built from simplicity, repeating itself into complexity. Carbon's ability to form four bonds, the hydrogen bond's subtle force, the flow of protons across a membrane. These simple principles, repeated endlessly, from the structure of DNA to the turning of the citric acid cycle, from the folding of proteins to the transfer of electrons, biochemistry shows us that complexity is not chaos but pattern, not noise but rhythm. Life thrives not by defying the laws of chemistry but by embracing them, weaving them into cycles and pathways that sustain continuity. There is a quiet elegance in the way biochemistry reveals unity across diversity. The glucose in your bloodstream is the same glucose that fuels a leaf, a fish, or a bacterium. The ATP that powers your muscles is made by the same processes in fungi, in trees, and in microscopic algae floating in the sea. The genetic code that writes your story is the same code that shapes the wings of a bird or the roots of a flower. Beneath the variety of forms, there is a shared molecular language, one that connects all living things in an unbroken thread. To study biochemistry is to glimpse that unity, to see beyond the surface differences and recognize that all life is kin. All life is bound by the same chemistry. It is also a story of cycles, the repeating flows that sustain both cells and ecosystems. The carbon dioxide exhaled in a breath becomes the carbon taken in by a tree. The oxygen released by photosynthesis becomes the oxygen needed for respiration. The sugars built in a leaf become the food that sustains an animal, which in turn returns nutrients to the soil. At the cellular level, molecules enter cycles, are transformed, release energy, and return to their beginnings, ready to turn again. These cycles reflect the deep truth that life is not a straight line but a circle, where endings are beginnings, and continuity is sustained through repetition. In contemplating biochemistry, one also sees the harmony between stability and change. DNA preserves information faithfully, yet mutations allow evolution. Enzymes catalyze reactions with precision, yet they can be regulated and adjusted to meet changing needs. Membranes create barriers, yet they remain fluid and adaptable. Energy is stored, yet it is always moving, always flowing. Life requires both steadiness and flexibility, both order and variation, both permanence and renewal. Biochemistry shows us that balance is not a fixed state but a dynamic process, a constant adjustment that allows life to endure in a world of change. Perhaps most quietly profound is the way biochemistry connects us to the cosmos. The atoms in our bodies were forged in ancient stars, and the energy that fuels us originates in the heart of the Sunday. Through photosynthesis, light becomes sugar. Through respiration, sugar becomes ATP. Through ATP, energy becomes thought, movement. Every heartbeat, every breath, every step is powered by this cosmic gift, transformed through biochemical pathways into the rhythm of life. To understand biochemistry is to see that we are not separate from the universe, but participants in its unfolding, carriers of its energy, arrangers of its matter, part of its ongoing story. Biochemistry also teaches humility. The molecules that sustain us are small, invisible to the eye, yet their work is unceasing and indispensable. Enzymes folding, protons moving, electrons flowing. These are the quiet workers of life, always at their tasks, never stopping, never seeking recognition. They remind us that the foundations of existence are humble, that great complexity arises not from grandeur, but from countless small acts carried out with consistency. The miracle of life is not in rare events, but in the ordinary, repeated endlessly with precision and care. As we conclude this gentle exploration, it is worth resting in the thought that biochemistry is not only the study of life at the molecular level, but also a reflection of life's deeper character. It shows us that balance sustains, that unity underlies diversity, that cycles connect endings to beginnings, that stability and change work together, and that small, quiet processes build great systems. To understand these truths is not only to gain knowledge, but also to gain perspective, to see life as both fragile and resilient, simple and profound, individual and universal. In daily life, biochemistry continues quietly, whether we notice it or not. It is there in the digestion of a meal, in the contraction of a muscle, in the release of a neurotransmitter, in the repair of a cell. It is there in the growth of plants, in the exchange of gases between forest and atmosphere, in the cycling of nutrients through soil and water. It is there in every organism, in every system, in every breath. Biochemistry is not confined to books or lectures. It is the living reality beneath every experience, the soft current that carries us from moment to moment. As this peaceful session closes, one can take comfort in the thought that biochemistry, though complex, is also deeply harmonious. Its pathways may seem intricate, its molecules countless, yet together they create a whole that is steady and enduring. To learn about biochemistry is not to burden the mind with details, but to open it to the quiet beauty of life's design. It is to hear the silent music of molecules, to sense the patterns that sustain existence, and to rest in the knowledge that within each cell, within each breath, the chemistry of life is unfolding gently, endlessly, and with quiet perfection. And so, as we step away from these reflections, we carry with us the awareness that life is chemistry, yet more than chemistry, that molecules sustain us, yet they also connect us to something larger. In the quiet turning of cycles, in the steady flow of energy, in the faithful copying of nucleic acids, there is a lesson that life is not only about survival but about harmony, not only about energy but about continuity, not only about molecules but about meaning. Biochemistry is the hidden song of life, always playing, always sustaining, always inviting us to listen more closely, even in the stillness of rest.